meters off bottom, almost there.
Good morning, deep sea explorers. If you're joining us today for this first dive on the New England Seamounts, we're on dive 12 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. We have just seen the bottom. We're going to give the pilot some time to set up, and then we'll officially start this dive. Thank you.
Come down a little bit more, and then I'll go ahead and get your swing arms out and ready.
Sounds good.
Are you comfortable with the normal configuration? Uppers out, lowers 45?
Yeah, I think for now that makes sense.
Understood. I'll come down to a delta of around 15.
Sounds good. Hey, Washley, just got to bottom. We're just going to take a few minutes to set everything up, and we will start the track soon.
Perfect. Thank you so much, pilot.
Enabling hydraulics, lower swing arms coming out.
Bridge, ROV nav. Yeah, I wanted to let you guys know that we do have the vehicles on bottom now, and ask if now is a good time to discuss bailout plan. Yes, we'd like to set the bailout to due east, 90 degrees. Good copy, thanks. Copy that, thank you very much.
Starboard upper is quite sluggish, huh?
Yeah, outboard it is.
Yep.
It's pretty good inboard. Go for stow. Blue view up.
Okay.
Okay, so steep, nothing too crazy.
Should be set up. I'm going to go ahead and come down a little bit more.
Sounds good. I'll put blue view out to, actually, Nav, can you range me out to about 50 meters, please?
Copy
For some
that.
reason, I keep losing control on this mouse.
No worries.
For your reference, I have you at 3.5, pilot.
Copy that. Lighting's all good?
Yes, lighting's good to go.
All right, Washley, I think we're all set. I'm going to take some looks around. If you see any items, I know we're looking for a rock. Looks like there's some in view.
I was going to say there's some right there. How about that? Yeah, definitely. First task is a rock. We'd also like a little zoom on the sediment at some point too, as well. It looks a little bit different from the previous sediments.
Yeah, of course.
Thank you.
All right, Biddy, you want to come in on rock real quick and we'll get some sediment? Video, you want to come in on rock real quick? Oh, that actually looks like it's cemented in, huh? Sticking out of the sand.
Maybe loose. It looks a little different from...
Yeah, it does potentially look like it's stuck there. Let's see if Kira is on the line. If you want to make a comment about this particular rock.
You could probably get both of them in the
Yeah, good morning.
same reach area.
It looks a
Go
little
ahead.
rounded, a little, I actually think it might be a cobble from somewhere else, maybe a drop stone. We could nudge it. There's also some sort of crusty material. It's hard. We just settled down, so it makes me feel a little bit interested in looking around a bit more.
Watch it. I think I heard to look around a little bit more.
Yes,
So,
yep.
Yep.
They're thinking that's potentially a drop stone, so it came here from somewhere else.
Yeah, copy that. Do you want to look at
Yeah.
sediment while we're here?
Yeah, let's take a peek at the sediment.
All right, video. You want to come on sediment?
I certainly see some of the pteropod shells that we were seeing in previous sediments, but not nearly as many as we were seeing before. Piece of sargassum there as well.
Yeah, and definitely some finer particles than on the shallower depths at the other seamounts.
Yeah, interesting. These pteropod shells, these little triangles and tubes that you can see that are bright white, and they're actually the tests of dead planktonic organisms called pteropods, sea butterflies that swim up in the water column.
Hey, Bob, can you come wide, please? Thanks.
Great. Thank you so much, pilot. So I think if we just kind of scope around a little bit and see if there are any more suitable rocks.
Okay, let me know. No problem. Yeah, next rock was this one right here. Do you want to take a quick peek?
Let's have a quick look at it. I wonder if that one's a little large.
Yeah, it's definitely bigger. I think that's within our range, though.
Okay. Yeah, let's take a look.
Video, you want to snap this rock? Lasers, maybe
Yeah,
25
likely igneous.
centimeters across. 20-25.
I would love to move a bit and search around to see if there's another rock.
Okay.
I think I copied that.
Yeah,
Just
thank
to
you,
give a
pilot.
little bit more
All right,
context.
video, come wide, please.
Thank you, though. Otherwise, a nice-looking rock.
It is a nice-looking rock. Kind of stuck up there from that platform.
Here's a side view. So this is to the south.
Yeah, kind of looking.
Yes, we landed maybe like on a 40-degree slope.
Yes.
Is it roughly that?
Yeah, that sounds about right.
So for those of you who have just joined us, welcome to Dive 12 of the NOAA Ocean Exploration Expedition,
kinda far
North
out.
Atlantic
Yeah, I
Stepping
was gonna
Stones.
say.
My name is Rian
I'd like to
Waller.
check
I
it out.
am the
It's a good
biology
spot
science
for a,
lead for this
a
expedition.
rock over there.
Kira Mazel
Yeah.
is on
I
the
don't really
telephone
see
line as the
too
geology
much.
lead. And
He
today
comes
we're
back
diving
and
on
then go
a
to
previously
port a little
unmapped
bit
and
and
unnamed
might find
seamount.
a
It's
ridge
been
with a
nicknamed
few.
by us the Y seamount due to a little feature up on its top that looks a little bit like a Y. And today, the seamount is made up of two peaks. We're diving today on the western peak, on kind of the eastern side. Today's dive is going to go up a slope, and we should hit a fairly steep wall about the midpoint in our dive, and then working our way up to a short plateau, and maybe a little bit beyond that as well. Because this particular area has been unmapped and unexplored before, we're seeing this for the very first time today, and so we're going to document biology and geological features along the way. And if you haven't joined us before, one of the first things that we like to do on these dives is we search around for a rock to collect. And I'll let Kira explain a little bit about why we do that.
Thanks, Rin. Yeah, we're looking to this because this seamount had been not previously mapped or dove on, or even any dredge rocks collected in the past. It's great to get a geologic sample right off the bat in the deeper flanks to try and sample the igneous geology. That helps so that we can get age dates and figure out how old this seamount was and how it fits into the long geologic history of this New England Seamount Chain. And so we're looking for places like here where we might be seeing some igneous rock flows, and we can grab a rock that looks like it's the building material of this seamount.
Yeah, and here we're coming kind of across our first larger of the fauna that we've seen so far. In the sediment there, we can see lots of fossilized coral remains, so little pieces that are sticking up and very black. Those are the stalks of corals that died probably a very long time ago. But here we have potentially our first live coral colony. We're going to zoom in here a little bit. It's a little bit of an unusual shape, so I'm kind of intrigued as to what this one might be.
All right, video will start to come in.
If you can see those red dots on your screen, those red dots are 10 centimeters apart. We use them to scale organisms on the seafloor, and this one is a fraud. This one is a sponge. This one is not a coral. It's beautiful. Really, really beautiful and unusual.
Hold that. Thanks.
So those laser scales are completely harmless. It's very difficult to tell what size things are on the seafloor.
Disabled joy lock instead.
And so we use these to be able to give us information on what sizes geological features are or what sizes organisms are on the seafloor. This is a really unusual sponge.
I wonder if it's an encrusting sponge rather than a like covered skeleton. Looks very coral-like.
Yeah. All right, video, you can come back in.
Yeah, it seems like usually there's an advantage to having a little bit more mass. I don't know. I think of sponges as being kind of more thick. It's curious to imagine why this one has such thin branching arms.
Yeah, absolutely curious. So these sponges are filter feeders, so they have all these pores that you can actually see really well on the zoom right now. And so they take in water and they extract nutrients and food out of it and then spit it out of larger holes. And so usually you think of sponges as being either encrusting, having large masses, or just being kind of these barrel shapes. And so this is a really unusual sponge. And I think this might be an idea actually to sample a bit of this sponge.
Oh, yeah, there is a big fish there.
Because this is something that's definitely very unusual.
Yeah, do you say sample watch it?
Yes, please, if that's possible.
Yeah.
If you want to visualize the fish, that's totally fine. We can come back.
If you don't mind, if we're going to stay here.
Yeah.
You are ramped up. Ship is stationary, correct, Nav? We haven't had any moves.
You can just move a little bit. I don't want you too far away from the slope.
Copy that.
Thanks.
Come to like a 260.
Yeah, sounds great.
All right. Have you there at 4.2. Decent visual. If you'd like, I can pull up Mini Zeus on my screen.
Yeah, if you're comfortable.
Yep.
You're nice and stable.
So the pilots here are going to set up to sample this really unusual sponge, very coral-looking sponge. And so we're going to give them a little bit of quiet so they can concentrate.
Seems very sturdy for a glass sponge.
Alright, try locked in. And watch it. I'm just going to grab what's closest to me. Does that sound good?
Yes, that's perfect. Thank you.
I think this is good. We can try opening drawer after. You might be able to get it open, maybe not. If you can't, I can just back up.
Yeah. Do you want to go to port, do you think, or?
Well, let me see how big it is. What's that snap?
I have Mini Zeus looking at your arm as you come live.
Sounds good. Arm's coming on in 3, 2, 1. Arm is live.
Copy that. I'll follow you over to the sample as you move.
Force 3. Dixing out.
Okay, I'll hold him Zeus there.
Sounds good. Hey, watch it.
Go ahead.
Video, can you zoom in on Mini Zeus real quick? Nope. Mini Zeus, please. Hold that. Is that enough, Watchlate, or do you want a little more?
No, I think that looks good.
Okay.
Yeah, that looks good. That's a good piece of brunch. Thank you.
Yep.
Yeah, that's perfect.
Okay, I currently have both bio boxes are open and ready.
Yeah, understood. Let's go to the starboard. I have
Copy that.
I'm indexed out.
Understood. Coming out.
Let me know if you can open it.
I'm opening now.
I guess let me know if you hit anything.
Looks clear. Keep an eye on starboard side. Okay, it did hit a little bit, but that's full wide, or full open.
Yeah, sounds good.
I'm
Okay.
going to come down with the Mini Zeus.
And then could you put the, let's see, upper port arm in, please?
Yep.
Just so I can get some more light there.
Okay, stowing upper port. Holding there. Do you want more?
That's perfect. Thank you.
Okay. So you may still have a piece on your jaw. There you go. Okay, I'm going to let that sink and then I'll bring it in once you get the arm clear.
Alright.
Okay, coming in with the...
There it goes.
Sorry, that's my fault.
No worries.
Indexed. Thought I indexed out, did not.
Okay, box is stowed. Save lane.
Alright.
I'm going to go ahead and bring your upper back out.
Sounds good. Thank you, Copilot. Watch it all set with that. Watch, I think I can't quite hear you.
Oh, I'm sorry. I'm talking back here to someone. Go ahead, please.
Is that all set here? Is that good?
Yes, we're all set. Thank you so much. Yeah, and just looking around, Kira had mentioned these
You might
rocky
zoom in on
outcrops
where
might be a
you
good
cut
area
it.
to find a fallen rock.
Copy that. I'll scope around.
Thank you.
Say again, Copilot.
I was just wondering if you could zoom on where you cut it. Get a sense of what the inside looks like, but just a personal. I'm going to go ahead and switch back to my
Yeah, sounds good.
monitor. And I am right at 270 still, so
Okay, that's great.
that's where we want to be.
There's one rock over here. I don't know if this is any better, but can look at it. Watch it. Is this rock of any interest? Should I zoom on it?
Let's have a zoom on it, but I'm going to ask Kira to kind of weigh in on the rocks. That one looks a little similar to the one that we zoomed in on that was a no-go earlier to me. But
Yeah, copy that. Video, you want to snap this real quick? Are we looking for something that looks like it broke off?
Exactly, yes. They're looking for something that's definitely from this area, and so, you know, looking on those underhangs where it might have broken off would be a good place to look.
Copy that. All right, video, you can come wide.
This
Should be
one
enough.
isn't too bad.
Oh,
I mean, it's at least in the company of many other rocks. It's a little bit more smooth.
Not too bad. Is that good enough for a sample?
The size looks pretty good.
is that a sample, Kira?
Yeah, I think we can. I think when we see one loose that's of a good size that might have some fresh igneous rock on the inside, it's a good bet. So let's go for it.
Okay.
Thank you.
Let's set up to collect this one, please, pilot. Thank you.
Right, pilot, you're wrapped up. We go ahead and switch mini Zeus back.
Sounds good.
Ship remains stable.
Cover that now. Thank you.
Let that sit for a second. And I'll just go. This looks pretty small, so I'll go starboard box. I'm sorry, not starboard. Port box with the lid.
Cover that.
All right, if you want to wing inboard.
Coming in.
Thank you, sir. Arms coming live. 3, 2, 1.
Port is all the way in. Disable. I'll follow you over with the mini as you come out. Holding there with the mini Zeus. Actually, I'll come down a little bit so I can get a... I'll come up a little bit to get the sample centered.
Copy that.
Okay, following you down.
All right, video, you want to do a quick zoom over here? Hold that. Do a little rotate in case we need it. Some items there. All right, video, you can come wide.
Okay, port's going outboard. Port wing is stowed. I'll bring the mini over to stow the arm. Holding there.
Our jaws are in. Indexed out. Arm is off.
Copy that. Setting up the mini for blue view.
Pilot, this is Watchlead.
Hey, Washlead Gal.
Yeah, we've had a request just to zoom in kind of on the sediment right behind where that rock was. There is something gray sticking up.
Yeah, of course.
Thank you.
Video, if you're all set, please zoom in. Thank you, sir.
can see in the background here is Xenophyophore. These are these kind of little half-ball shapes with lots of little pits in them. And I think
there.
the
I'm going to switch back to my
zoom...
camera if you're ready.
Yeah, go for it. Thank you.
Copy.
Others I think might have come in here and made some holes. Maybe up a little bit, if we can.
Yeah, tilting up.
Yeah, beautiful shot of that Xenophyophore, for sure.
Holding there.
These Xenophyophores are single-celled organisms, and they make these houses of mucus that take in these sand grains. Thank you so much, pilot.
Copy that. Lasers there for reference. Looks good. All right, watch it. Anything? Do you want me to keep looking for under crust where rocks potentially broke off?
No, we're good for our starter rock, and
Okay.
so we should continue on our line.
Sounds great.
Thank you so much.
Yep.
All right, pilot, I'm still at 270.
Yeah, copy that. All right, I say I'll do a couple passes and we'll keep going up.
Let me know when you're ready for a ship move. Standing by.
Yeah, copy that. I haven't explored the starboard side yet. I'll just take a peek, see what we got. Let's see. Here's camera 4-2. Looks good.
Really good returns on the blue view.
Yeah.
Lots of these sediment ripples here, Kira. This is really, really interesting. We had a question earlier in the chat room about how these sediment ripples occur here in the deep ocean, like in the up in the upper ocean. You can kind of understand all that wave action, but how do they actually occur down here in the deep?
Yeah, that's a really good question. I mean, it ends up actually being similar processes. We think about the deep ocean as being completely still, but there are actually still some currents that reach down this far, and so you can still get sediment ripples forming at deeper depths from the currents moving it and creating these little ridges. And the sediments, when they have any movement like that, they can get sort of size sorted, so you can get different grain
just start with
sizes
15 meters.
building up
anything.
and then having the other grain sizes in between and creating these little ripple
slope.
features.
I don't see any cliffs or anything.
It's
ahead.
likely still from
How do you feel about that?
currents passing
Yeah, we can do
over
it.
slowly over
18
time,
meters,
creating these little
270
sediment
sounds
ripples
270. Decimal 2, okay.
without much
Yes,
disturbance
Copy that. That sounds good.
otherwise, so allowing the
Bridge
sediment
RV
ripples to form and stay in place.
Yes, we'd like to request a ship move, please. Range 15
Yeah, and it's quite
meters.
extensive here as well. It's kind of in
Bearing
between these little
270
plateaus, but also up on
degrees
top
at
of the
decimal 2
plateaus
knots.
as well. These little kind of rock ledges.
Yeah, this is a really
Good
interesting kind of
Thank you.
for these little crusting outcrops. It definitely looks like there's ferromanganese crusts
Bring
on
my
them,
repeater to a
but what the
3.5.
underlying geology is
copy
kind of obscured by the sediment cover and the ferromanganese crust cover. Doesn't seem like at this depth we'd have a lot of carbonate, so these might be little ledges of igneous rock covered in crust, or it's a really neat morphology.
Yeah, it's interesting to think about
Certainly
the...
since they're
Oh, go ahead, Kara.
Oh, sorry, I was just going to say, since these are a little bit more covered with sediments than we've seen in other places, it seems like there has been some sediments cascading downward, or there have been some periods of higher sedimentation rates to sort of coat the upper portions of these crusts with.
Oh, I think we have a little crab under a rock there to the right, pilot.
Yeah, copy that.
Yeah, it's interesting thinking about those currents, that created those ripples. There was an interesting conversation here in the scientific chat room about that sponge that we just collected and how unusual it was to see sponges that are in that coral shape. And it was mentioned that, these sponges have been known before to be in these coral shapes, so they tend to live in areas where there's not a huge amount of water flow. And so they can get away with being kind of thinner and getting
crab.
water flow kind of all around their branches rather than just against one face. So it's interesting to see those ripples where it shows a bit of current, and then to see these sponges that might not necessarily like that kind of current, though maybe they were in a slightly lower location than the ripples themselves. Interesting. It's a Chaseon crab that's hiding here under this rock. Looks like it's been in the wars a little bit, with its claw broken off at the tip there. We were told earlier in this expedition that these crabs do regrow their legs and their arms, and so it will eventually grow back.
All right, is that all you need, Washley?
Yes, that's great. Thank you. You can see all those pteropod shells in this particular area as well. You know, on the sediment when we zoomed in earlier, there weren't many, but here, kind of around this rock here, where the current must have accumulated these shells in this particular area. Oh, if we could zoom. Oh, actually, no, I think that's a dead coral skeleton. Sorry, ignore me. This is quite the ledge that we're going over here. So we are currently at just over 2,800 meters, too, so we're quite deep at this point.
Go ahead,
Yeah, and
Rich.
Kevin Conrad adding from the chat on the geology side that these look like flow tops of igneous
Watch it go.
formations with the ferromanganese crust growing on top.
Yeah.
Usually crusts don't like to grow in conditions that are this sedimented, but you can have, over the geologic history, you can have different sedimentation regimes. So there likely were portions when this seamount was covered in less sediment, and those crusts could build up, and now they're being coated with a thin layer in the current oceanic environment.
Swinging, but you have plenty of time.
Copy that. All right, video, you want to come on in?
Just gonna have a little peek at this ledge here and see what's living around it. Certainly see some potential hydroids, some of these large forams, which are branched like. Oh, maybe a little shrimp or something hiding under there. I just saw dart away. Oh, and some tube worms. You can see on the bottom right of our picture here, there's this long tube with kind of this feather sticking out the top, and those are tube worms, and that's kind of their feeding tentacles in this fan shape here. A little snail at the top as well.
Ooh, attractive.
Yeah. Oh, and a fossil coral. Look at that. There is one.
Kind of pan it around, watch it.
Yeah, that's great. I thought I saw something pink up to the left of the pan too, when we kind of come back in that direction.
Yeah, video, come out a little bit.
It might have been like in a ledge just up from this very base one.
Am I going the right way?
Yep. And there's some stalk sponges here as well. I can see some brittle stars. Yeah, actually, I'm not sure if we're going to be able to see it. So it's in the lasers now.
Oh, down there.
Yeah.
Yeah.
Might just be a bad angle, but I'm wondering if that actually might be a little cup coral or an anemone under there.
Hey, video, go in. Can't really get lower from this position, but
they never pick the easy places to settle. Yeah, I think this might be a little cup coral, really hiding here, right under this ledge. These cup corals like to live in areas where water is slightly enhanced, so it might go a little faster, because that brings in food and takes away waste. And the reason we can see this as a cup coral is you can kind of see the stripes of the skeleton. It's very, very orange, but you can see those stripes of the skeleton and those tentacles sticking down. Thank you so much, Pilot.
yeah, absolutely. All right, video, whenever you're happy.
And just above it here, on the left, there was a crinoid.
Do you want zooms on that?
Beautiful. Sure, a quick zoom would be great.
Right,
I think
video,
it was...
snap crinoid real quick.
These crinoids are often quite difficult to identify. Oh, actually, no, maybe that's not a crinoid. Hang on here. Yeah, if we could get a good zoom on that one.
I'm at max zoom.
Alright, video. Try to keep it center.
That's where I'd love some input from our scientific chat room. So,
Is that all the zoom we have?
yeah, black coral. Has those very black coral tentacles.
Roger,
Not
that's
seeing
max.
much of a skeleton in there, but boy, super fine. So Tina Moltzover from Russia is saying that this is a heteropathes. And actually, I'm not sure this is one that I have personally ever seen before. It's really beautiful, very unusual shape. Kind of this star-shaped.
Alright, watch lead. Is that good for that?
Yes, thank you so much.
Okay.
Yeah, so saying this is potentially a heteropathes opresci.
Got it.
And we're kind of, Scott Francis is saying we're looking down from above, so it's kind of the star shape, and it must have a stalk underneath there. Beautiful.
I have not changed my camera angle, so I'm still looking down 3.5, so you do have time.
That was a really nice find.
And my head has come over a little bit, so I'm going to bring it back to starboard, back to a 270.
Yeah, sounds good.
So if you're just joining us here today, welcome to Dive 12 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. We landed on the bottom about 30 or so minutes ago, a little bit more than that. And we are currently on Dive 12 on a previously unmapped and unnamed seamount that we nicknamed the Y seamount this morning because a little Y feature up on its top. We are diving today at 2,800 meters and working our way up a slope to a steep face and then up onto a plateau, hoping to reach about 2,500 meters during this dive.
Alright, Nav, when did that last move end?
Last move
It's been very
ended at
interesting so far
14:33, 10
in
minutes
this
ago.
landing spot compared to the past three dives that we had over in Corner Ice Seamount. Now
I
we're
suspect
seeing a
Sirius
lot
is
more
stable.
sedimentation, which
Yeah,
I
Sirius
expect
is
will
stable.
increase as we go
Sounds
in
good.
towards
Definitely slope. I
shore
don't see any walls.
on each
I think it's
seamount
about
as we
another
continue
15.
to head
270.
west
That work for you?
back towards the
Sounds
east
good.
coast.
Now.
I'll put it
And
in.
we're seeing these finer grain sediments, a lot less coral.
Bridge
We're
over
also a
nav.
lot deeper today, so if you're,
Yes, another ship
if
move,
you're
please.
curious why what we're seeing is a bit different
Same
from
as
the
the
past
last.
3 days,
15 meters range,
that's why.
bearing 270 at decimal 2 knots.
Seeing these rippled sediments and crusty outcrops of ferromanganese crust coating igneous flow.
Copy that, thank you.
Yeah, absolutely, Kira. So at these depths, we'd expect to see more in the way of kind of black coral fauna than necessarily the octocorals or the scleractiniums. So it was really nice to see that little cup coral that was hanging out under that ridge. And then certainly sponges. So you can definitely see on the seafloor here, you can see these stalks that are sticking up of old colonies of coral that have long since died and left behind these stalks on the seafloor. The changing environment a little. There's something pink kind of dead slightly to the right of center up on that ledge there, pilot. It's possible to have a quick snap zoom.
Yeah, copy that.
We're seeing some sponges as well. I actually made a collection of a slightly unusual, potentially new location for a
Is that what you're
sponge.
looking at, Watchley?
Yeah,
That's
that's
Do you want to snap real
actually
quick?
slightly left of center, kind of up on the ledge. I think it's a persingid. There we
Okay.
go.
I was looking at a rock.
Not that we don't like to look at rocks.
Yeah, I thought it was something else.
Oh, this is beautiful. So this is a persingid sea star kind of wrapped around one of these old stalks that I was just talking about here. And these persingids like to be up in the water column. They are suspension feeders, so they pick particles out of the water column to feed. And so they like to be up high. And so this one's climbed on top of this this stalk here. It doesn't have any of its arms really out in the water current feeding right now. but sometimes we see them with their arms really stretched out very wide. This one's got a beautiful disc to it too, this kind of central area here. Wonderful. Thank you so much, pilot.
Yeah, of course. Lasers for reference.
There's a coral actually right behind it. Slightly left.
Right there?
Right there. If we can have a quick zoom on that one.
Sure. I'm far, but videotape it.
Okay.
I mean, it should be enough for ID. I can always get, it's in our track, so I can get closer.
Just looking at this coral here. This looks almost plexaurid-like, though. I know Scott France and Tina Moltsover in the chat room here. Scott says potentially a corallium. Yeah, a corallium. Actually, growing off of this old stalk here. Thank you so much, pilots.
Yeah, copy that. That video. This laser's there. Looks good.
Go ahead, Bridge. Copy that,
This
thank
is a
you.
very rugged terrain. We're seeing, I think, a lot of ruggedness coming
Ship
from the
move
rock
complete, pilots.
below, the igneous rock below,
Copy
as
that,
well as
Dave. Thank you.
lots of ferromanganese crusts that are coating the flanks of
This
this
looks like a good one.
igneous flow. Definitely a totally different landscape than I've
Looks
been
like
seeing
you might
since I
have a
took on the
row,
ship
yeah.
several dives ago.
It's a lot
Lots
good
of, here's a
items
little
coming up, huh?
sediment
I'm
accumulation
gonna
in
lift up my
between
camera a little bit.
and some
Get a
more
look out in front of you, if you don't
plate-looking
mind.
rocks
of course.
underneath this big boulder. Very interesting.
Yeah, these
Pass
Thanks,
those
video.
items.
big boulders have definitely caught my eye. They seem to have a lot of organisms living up
Yeah.
on top. It'll be exciting to take a look
Don't
when
really
we.
shoot too much.
when we
I'm gonna
get
come
there.
back down and
But yeah, definitely different. You know, we
Enjoy
just come out of
35
those
again.
sediment ripples, and
Yeah,
already
sounds
we're in
good.
a very different geological morphology and different habitat for organisms to live.
okay, that's 35.
So where we see boulders sticking up like this, these are often places where we find a lot of life, filter feeders and suspension feeders. So those organisms that want to take out particles out of the water, and these boulders can actually kind of enhance currents, so they can stop the flow of water and kind of move it quickly around them, creating areas where there's a little bit more current for organisms to grow. And so what I'm seeing here. Is actually primarily those sponges that look a little like
Looks like a
corals.
long-legged sea star at the bottom.
And Scott France had sent us a picture from the Pacific, a
lot of stuff
Stylodorix
here.
species sponge that we wonder if this is.
Right, well, actually, do you want, seems to be a lot of items in view. Is there something specific you want to look at?
Yeah.
I'll just
Oh,
kinda,
so many choices.
now.
Actually, there's a coral on the right-hand side. It'd be good to get a quick snap zoom on, kind of purpley pink. And then we can take it over the other side. I think most of those white are actually these sponges like we just made that collection of.
Is this, as
Yeah.
I center up? Is
Yep.
that what you're looking at?
Yeah, I think we're aiming for the same one there. Just an ID zoom would be good here.
Yeah, copy that. Alright, video, you want to snap real quick? Not a great angle for you, huh?
Not a great angle, but this does look a little like a corallium as well, which is a type of octocoral,
Video come wide. I'll come up and look down at it. Might be better.
and one that we do sometimes see at these deeper depths. So we are currently at 2,793 meters deep.
Alright, video, come back in.
There we go.
Is that better for ID purposes?
That is, yep. Yep, that's much better. Yeah, I think this is a corallium here. Thank you so much, pilots.
Yeah, copy that.
Interestingly, there's a bamboo stalk down below, but it's all the tissue is eroded away, so I wonder if we'll see some bamboo corals as we
Alright,
work our
video,
way up.
come wide.
Scott France is noting he had seen that Pacific dive from the Okeanos Explorer where they found this stylodoric sponge, and he says it's a very, very similar pattern, really, really dominant on these boulders that are sticking up. A fly trap anemone up the top there, and a really large sea star here hanging over these old dead branches. There's a couple of other species of sponge in here as well. These ones on stalks with kind of a wider face, more traditional sponge looking. I think this is another Bersingid sea star, just a really, really big one, very, very long arms.
It's interesting how on the right side of this giant boulder, it seems to mostly be dead things, and then most of the life is kind of on this ridge feature at the top of the boulder and not on the other side.
It is, yeah.
Maybe that's the best location for the filter feeding.
Yeah, you must assume that this location is just best for those larvae, however many years ago that was, to settle here and grow. You can imagine kind of on the right side, it's maybe less of an angle. Maybe it accumulates a bit more sediment. Maybe that doesn't make it quite as pleasant to settle as a larvae. But it is really interesting to see them kind of really colonizing very densely this ridge at the top of this boulder.
Almost like a mohawk made out of coral.
It is, yes. A little sponge mohawk. Thank you so much, pilots. That was some great video.
Yeah, no problem. GoPro, I'll take a peek.
And thanks for that correction on sponge, not coral.
Yeah, that's okay.
Yeah, definitely a big boulder field like that with lots of rocks. It seems like it could be a mass wasting deposit, which is when you have a lot of downslope debris from higher up building up and creating sort of a rubble field. And now on the other side, we're seeing this kind of sediment-rich morphology where maybe some of the sediments have built up instead of on the other side where we had a lot of rock buildup. Definitely seems like there's a lot of downslope movement. And as we looked at the zoomed-out image of the map that our mapping lead put together for us, we definitely knew that diving here we might see some slumping and mass lifting downslope of material. So it's neat to get to see that after looking at the maps and choosing the dive site.
Yeah, absolutely. And if at home you go to live stream number 3 right now, in the bottom right-hand corner, there's a little screen called the Blue View, and this is a sonar that our pilots here are using to help guide them on this dive. It tells us what's coming up a front. And every time you get kind of a bright light, it's a hard return, so meaning it's a hard area of rock. And you can really, I mean, it's a wonderful view right now in the Blue View. You can just see all these boulders and rocks from this potential mass wasting event here.
All right, Nav, when did the last move in?
At 14:47.
Copy that.
If we could have a zoom on this, I don't know if it's alive. These white skeletons that are sticking up here look quite fresh.
To the right here?
Yeah, just to the right of the lasers, really long. Thank you.
Video, let me get a little bit of a better angle. All right, video, you want to come on in?
So this is really interesting. So far we've just seen some black corals, and we've seen a single cup coral. But this is a bamboo coral, and they're called bamboo corals because they have these nodes. So the white skeleton here that you're primarily seeing is a calcium carbonate based, and then you can see these little ridges. These nodes are really tiny. They're really tucked in there, but they're often a black or a dark brown color, and they're actually made they're proteinaceous. And this one, I believe, does have some live polyps right at the top, but it's a primarily dead coral. Most of the skeleton is bare or encrusted with other organisms. We have here, I can see some foraminifera, these large branching. There's potentially some hydroids here as well. If we could scan up to the top pilot, that would be really helpful, and see if we can see those live polyps that were up here.
Yeah, of course.
Just barely hanging on. There's a snail with its proboscis sticking out there. Actually, as we go up, you can kind of see these.
Sorry, co-pilot. What was the last thing you said?
These nodes.
I was just saying that by
There's
swinging his
just
stop.
a few alive
like
right at
you're
the top there.
looking for another move, but we'll wait until they wrap up here.
I think we could start 15 meters if you guys are good
Yeah,
at
you
that.
think so? We can't really see anything behind this. I can look up.
I can hop up after this. I had peaks on my way in. It
Okay.
looked good to me. Well,
Alright.
similar.
Would you like to take a look before we move, or put it in now?
Say we
Yes,
can
I would
start
say that
and
this is
decide
likely to
mindful
be.
after we come up and out.
This is
I'll
not
put it
another coral that's come and landed on this particular
Bridge already
coral. It
now?
looks like it is part of the original colony of this bamboo.
Hey, Bridge, another ship move if we could. 15 meters,
Scott Front
range.
saying that these
bearing
nodes that are
270 degrees,
barely visible
decimal
could
2
be
knots.
that they're overgrown, or that the protein is actually degraded from them as this coral has died.
You got it, thank you.
If we could have an overview of the whole colony, that would be great, pilot.
Yeah, of course.
Thank you.
That video if you want to go for a full shot.
Kind of looking around this colony, see whether we can see a sea star or other predators that might be on, and might be a reason for this this coral not being in such good shape. See, there's another live branch over to the right there as well. It's interesting though, I really don't see
Stand
any
by, I can't hear
major
you.
predators on here.
There may be another coral down there at the
Oh,
bottom.
copy.
Large branch that's broken off there to the left as well.
Different from what we've seen. So
Is that
Interesting.
what you wanted to watch, Lee?
Yes, this is great.
Okay.
Yep, there's a chrysogorgia over to the right as well.
Okay. Is that all you need, watch, Lee?
Yep, that's perfect. Thank you so much.
Video if you can come wide, or is that wide?
That's wide.
The contrast of those ferromanganese coated stalks in black against the
All right.
bright
Go
white
out,
of that
little
barely
rise.
hanging on coral is just beautiful
Copy that.
image there.
I should start swinging here shortly.
Yeah, copy that. So we're seeing back there 30 meters, something right there, which is probably just these rocks there. It's probably the high point. So I think that looks pretty good. No sheer cliffs or anything that I see. Just looks to be more outcroppings.
This is some really interesting terrain. These large boulders, kind of as we're coming up to, as we're starting our way up the slope. Some more of these sponges, look a little like corals. It is interesting to see that some of these boulders have these massive cracks in the top as well, kind of fractured.
Yeah, I was just looking at all of those fractures. It's kind of difficult, I don't know, to think about how you can break igneous rock like that, perhaps when it was tumbling down to this location, or some other rocks have fallen on top. It could be cracks in the manganese coating on top of the rock. When we see a debris field like this, it's interesting to try and imagine how long ago this mass wasting event happened, where we have a lot of material flowing downhill and creating sort of an underwater landslide. We can get clues based on how thick the ferromanganese crust is. You know, if we had a lot of exposed surfaces when the rock broke away and fell down to this location, then you could have a thinner ferromanganese crust growing on it, which grows about 1 millimeter per million years. And so you can get some clues about how long the rock has been sitting there from how thick the ferromanganese crust is on each side, which sides may have been more exposed to currents or more recently broken.
This is a really interesting boulder up on top. It looks like there's quite the sediment accumulation on this kind of flat face on the very top. And the sediment in the background, there must be a fair amount of sedimentation rate in this area, or it coming down from above, I guess. You can see some more corallium here and some more of these sponges. Yeah, sorry, Kira, go ahead.
Nope, go ahead. I was just agreeing with you that it looks like a pond of sediment.
Yeah, right on top of this big boulder. It's amazing me, too, all of these old skeletons that are sticking out of this boulder here. You know, at these depths, this must have been an area for certain species to have really thrived at one point in time. And we don't know how long ago that these particular coral skeletons that we're seeing that are covered over in the black, we know that they're covered over in this ferromanganese patina, and so they've been there for a very long time. Other hard corals, little cup corals that have been dated from this kind of region, a lot of them lived 10 to 20,000 years ago, but some have been dated up to a couple of 100,000 years ago as well. So some of these corals are really paleo corals that lived here a very long time ago.
That's for reference. I'm
looking up quite a steep slope here.
Looks like more rocks, about 15 meters out from D2, so it's about 30 meters from Sirius. Looks like I'm seeing past most of them now. I don't see anything that looks like cliffs. Looks good. Explore this side.
If it's possible to zoom in on the coral colony on the left of this large boulder coming up, that would be great.
Yeah, copy that.
Thank you. Again, this boulder kind of following that pattern of having a lot of organisms living kind of on this top ridges and faces, being a little bit more bare. And these are possibly areas where the water current is just enhanced that little bit on the top, whereas the sides, maybe they accumulate a bit more sediment. This might have been an area that was just better for larvae to settle, better for larvae to survive predation as well. We don't really fully understand all the factors that allow organisms to grow on the seafloor in the deep ocean. This looks like another chunky bamboo skeleton here, but nothing is currently alive on this one. This one's been dead for a while. It's very overgrown. You can see those nodes. Yeah, that one's been there for a long time.
Is that all you need, Watch?
That's perfect. Thank you, pilot.
Video.
Oh, right below it on the left. I don't know if we're in a good position. There's kind of this white stick sticking out here.
I can reach that.
And I think I see some polyps. I think that one is still alive.
Yeah, I have a little bit of swing, but not a ton left. We should be fine. I still have you at 3-2.
Copy that.
Oh, a little underhang here. One large boulder resting on another.
All right, Vidya, you want to come on in?
Yeah, these are some massive boulders.
They are huge. They are absolutely huge. Oh, that might be a black coral and a bamboo. Very small amount of this particular bamboo still alive. And then this white colony up here definitely seems to have those very...
Morning, Reen.
Oh, there's Scott. Good timing.
Hi, how are you? I've been following along, watching these really old, ancient, mostly dead bamboo corals, and I was hoping this smaller one down here was going to be a fully live one, but it's also not doing great.
No.
A whole lot of tissue.
Not at all.
And as you noted there,
Yeah,
it's got like one branch of polyps. We'll see if it can hang on. And then, yeah, I think that was a black coral just above it, that fuzzy white yeti thing.
Yeah, I believe so. Pilot, is there any chance of zooming in on the fuzzy white if we're in a good spot to do that?
Is it the one still center?
Just up from center.
Up from center.
Very white, very fuzzy.
Is it, as we zoom in, is
Yep.
that
Right on
direction?
on the lasers there. For those at home, those lasers are 10 centimeters apart. They help us to tell the size of organisms on the deep sea. Yeah, that one's a black coral, Scott.
Yep, I agree. It almost looks like a Storopathes that has been bleached. You know, normally those are pretty bright orange in color, but it has the right branching pattern and the right polyp shape. We'll see if Tina Molodtsova from Russia, who's an expert in this group, has any comments on the matter.
Yeah, interesting.
Looking at the back of the colony, you can see that a lot of these black corals seem to have one face, and the polyps are sort of facing away from us. And so we're seeing the back where we can clearly see the skeleton very well. Tina does not believe it's the genus Storopathes, something else.
Yeah, interesting. And so the...
Maybe, Tina?
video come on?
Thank you so much, pilots. That was great.
Yeah, is that all you need?
Yep. Yeah, and so those black corals, that one is obviously white, not black, but they're actually named for their skeletons. So their skeletons are a very dark, very deep, almost pearlescent black color, very often very shiny. And so a lot of these corals have been named from the skeletons because they were collected from the deep ocean by more traditional methods such as grabs and trawls. And so very often a lot of the live tissue would have been sloughed away, or they would have been dried on board research ships and then identified and described from their skeletons.
Okay, but I'll center up.
Another aspect of the skeleton that's very
explore
interesting
what
is
this
that they're very spiny.
it's still
This
pretty
group of
much
corals
unwrapped.
is also sometimes referred to as the thorny corals.
Looking down to
And when you put them under a
3-2,
microscope, and
15.
especially a scanning
that.
electron microscope, which
I'll
can
come
look
up
at
as you
real
do.
detail at these skeletons, you see just an
good.
amazing diversity to the morphology of the spines themselves. So some may be curved, some may be upright and straight, some may be bent towards the skeleton, some may even bifurcate, their lengths can change. And so all those aspects of the spininess and the thorniness and how spiny they are on first, second, or tertiary branch
then do you wanna
go
maybe
into
center this
the
up?
definition
It might
of
just
what
be more
the
rocks,
species
but
is and how to describe
might
it
be
and
the
how to
most interesting
identify
path
it.
to follow.
And
Check that,
it's
coming
one of the
over.
challenges for us in trying to identify these things in video is that what we're used to is looking at the characteristics under the microscope of the skeleton. And so as we develop this technology for exploration and seeing more of the deep sea and seeing more of these animals in situ in their natural habitat instead of, under a microscope or in a bottle, we're sort of
It's a
trying
little different.
very hard to transfer that information from what we know in the lab to what we see in front of us
Back
so
up
that we
and
can
sink.
better make an identification of what it looks like in its natural habitat. Not sure if that was a worm or a fish.
That was a fish going by. Yeah, we can just about see it here.
It's the back end, at least.
Back end, I don't know if we can get a look at the front end here. It definitely has that cusk-eel type shape, I believe.
You just kind of turn it away as I rotate.
Refusing to show its face. A lot of fish in the deep ocean are very, very slow moving, and that's really to conserve energy. Now in the deep sea, there's not much food at this depth. We're currently at 2,779 meters, and so most, if not all, of the organisms in the deep ocean really try to conserve as much energy as possible. This one has some very interesting coloring on it. This very dark.
Come in a little bit, partial.
This really dark coloring around the fins there.
Video, you want to come partial? Hold that. Thank you.
Very, very white and bulbous head on this particular fish here.
It's coming towards me now.
Yeah, that's a good zoom if we could hold that. We have Peter Oster, who is an expert in deep sea fish, on the scientific chat room. So each dive that we do of this expedition, it's not just me on board the ship, but it's also a team of scientists who are working in the background to help us identify organisms on the seafloor, particularly where we find very unusual ones or things that we might not have seen before. It's one of the great benefits of telepresence, is being able to involve the community beyond just us on the ship here.
I'll let him swim out of here.
Very unusual face there. Yeah, it's actually got Peter Oster a little stumped as to what that might be. That was a very unusual looking fish.
Oh, actually, he swam out of view.
That was great. Thank you so much, pilots, for following the fish.
Co-pilot, look back to where I was.
Copy that.
Yeah, still checking out this really varied boulder field from a mass wasting event, seeing lots of igneous rocks that have tumbled down slope from up above, and the sediment that has either slid down and accumulated or continued to accumulate on the from up above through the water column.
So I came over a little bit,
showing us this really varied rocky landscape.
Yeah, I'm coming back to pour a little.
Just a hair.
It's really interesting to see lots of fossil coral and sponges sticking out, indicating that at some point this environment was much more conducive to more dense communities, and now a lot more sparse.
If there's anything you can do to try and
so it's interesting to think, has the sedimentation rate increased, and now there's too much or not enough food. These big boulder outcrops would typically be a lot
still
more
70 meters or so before
attractive, I would think.
Indicates a steeper rise.
Yeah, copy that.
But
I don't see anything.
Keep our eyes open.
Definitely more rocks like we're seeing. I don't see any potential walls. It's 15 meters out. Let me come up a little bit and look.
Copy that. I'll follow you up.
So we have this 15 meters
seeing
out.
These really large rocks with lots of smaller rocks that have been broken up as they typically fall down slope.
It's that right there. Yep.
Sediment accumulations, definitely, as you can imagine, on the mountain, sort of a landslide going down slope. I think that's what we're looking at here.
Nav, when's last move over?
Last move was over 14 minutes ago.
Okay.
Do we want to change the direction to like a 285?
Yeah, I think what you're looking now looks pretty good. Is that all right, Nav?
285?
285?
Yep, 285 will work.
So there's been some debate in the scientific chat room as to what that unusual fish was, and it's come down to an old fitted type fish from Peter Oster here.
Range will be 15 meters
Not
again.
quite sure what species exactly, but some great
degrees,
camera work there.
decimal 2 knots.
Zoom in on that fish.
Thank you, Bridge.
Pilot, this is Watch Lead.
Hey, Watch Lead, yeah?
Yeah, can we zoom in on kind of the bottom of that boulder central? There's a coral orange kind of twisting in the current there, over to the right slightly.
Yeah, copy that.
I'm bringing my camera back down to 3.5.
Confirm.
Copy. Right around there? Is that Where are you looking, Washley?
Yes, lasers are right below it right now.
All right, Biddy, you want to snap?
Yeah, this looks like a Bathypathes, another type of black
Pretty
coral.
far
We've
from
seen
it,
these
but-
actually fairly commonly. Oh, you can see beautifully the two little polychaete worms that live in the spine there sometimes on this particular species.
Back center.
Has those long, drippy tentacles that are often indicative of these black corals.
Do you see an ID for it, Watchley? I'm kind of far from
It's
it. now.
great. Thank you so much.
Okay.
Yep.
This might actually be a place to consider a second rock collection, although we're still pretty deep. Looks like it could really be igneous, fresher igneous rock that's flowed down slope, and it has a thick ferromanganese crust.
Pause ship real quick.
Do you think,
I think
we could
this is okay.
Bridge,
take a
could
look
we,
and see if
this is RV
there's
nav.
something of the right size
Could we come
in
to an
this
easy stop,
debris
please?
field here?
Thank you.
And then just find out how much that move went in.
Would you be able to, Bridge, also approximate
Yes,
how
the
much of
pilots
that
are
move
just
that we
figuring out if
completed?
this is a good place to set down and see whether we could collect a rock
7
here.
meters, good copy, thanks.
Should we
We'll go with
just a minute to
Yeah,
see
that
if they
should
can
be
do
fine.
that?
I have you at 35, so. And you're also going to be coming down a little bit to get a foot in,
Yeah, sounds
or a
good.
toe in.
So Watchley, this is a good spot. Just dead center, something in here. Is that?
Yeah, I should
They
let
would want?
let Kira guide you a little bit here, but usually angular is a feature that they often look for.
Okay, understood. Get in, set.
Going to come down a little bit as you do. Maintain that 15
Yep, you got it.
meter delta.
Angular.
Can you bring in my lowers inboard,
Oh,
please?
it sounds like we might have just lost Kira, but it sounded like angular was
you're fine,
the
but
thing
yes,
to
I
go
know.
for.
Lower is coming inboard.
I'm sorry.
Sorry,
stow
stow.
in.
Understood. Stowing. How do you feel about the uppers? Do you want me to tilt them back, fully deployed?
No, I think that's okay.
Copy.
Thank you.
Okay. When you're stable, I can bring up Mini Zeus.
Yeah, I think it's good. I'm gonna
Bring her up. And then coming down and look at the sample.
All right, Watchley. So I'll just kind of poke around up front. Does that work?
Yeah, absolutely looks good. So the request is for kind of angular and flat. Angular and fat, not flat. So
Angular fat. Understood.
yeah.
All right. Arm is coming live. 3, 2, 1. Arm is on.
I'll follow you out as you reach out. Angular and fat.
Just start low. This looks cemented, but moving a little bit there. All right, it's stabilized.
That one might have moved a little.
Oh, did it?
Thought it.
I think you may have moved a little bit when you
vehicle
pushed off.
definitely
It did. Okay.
I'll try again though. Yeah,
No.
that's nice and stuck.
Okay.
There.
There was a suggestion for the one kind of dead center just below the lasers. I don't know if that one is loose.
Was it the first one I touched?
Oh, did you touch that one already? I'm sorry.
I think right.
Yeah, that one right there.
Yeah, I think it's nice and
That
cemented.
one's not going anywhere. Okay.
What about the one slightly to the right of that? You can see it right here. I think it's that kind of long, skinnier one. Down the lower right-hand corner of your screen.
Down there.
Yeah, right underneath the jaws now. Yeah. Looks like it might be resting on a few of them. Is that pretty cemented as well?
Yeah.
Huh. You may have a few more options up slope that may be slightly out of your reach. I can bring up Mini if you'd like.
Yeah, maybe rotate up a little bit.
Okay. I'll hold there. Bottom part of that may have shifted. Or not.
So. Here, why don't you pan down a little bit?
Coming down.
I'm just gonna hold that.
Holding.
Actually, do you want me to try up top? It all seems kind of cemented right here.
Yeah, it's interesting. We're not quite sure why it's stuck quite like that. Yeah, if we could have one more try up a little bit, and then if that doesn't work, we'll keep on moving.
Yeah, understood. I'm just gonna adjust a little bit, get
Thank
a little closer.
Do you want me to bring Mini so you're looking at, like, out in front of you?
Yeah.
Because you are getting a little close to that, right here. We're pretty close to this rock.
Yeah. Can you point it up a little more?
Coming up.
That's the, yeah, right there.
Okay. Do you want to range in the blue view as well? Way down to like 5 meters? Does that help at all? Or?
That's right. I have good visual on everything. Thank you, though. So an inch a little bit closer. That's good.
You ready for me to come down?
Yeah.
Come down.
Yeah.
I'll look at you draw and then follow it up as you come up.
Sounds good.
Holding there for now. There we go.
That one's loose. Is that okay? Watch it. Did you see the one I just touched?
I did. I'm just waiting for a bit of feedback from Kira or from Kevin about this boulder. It's kind of directly under and slightly to the left of the manipulator right now.
Video, you want to come partial on main HD, please? Yep. Hold that.
Since it moved, it seems could be worthwhile. It is fat, definitely some material inside. It's not quite as angular, but it looks good to me, and Kevin agrees. So let's go for it. If it's grabbable, let's do it.
Perfect. Thank you.
Stand by. Might have got 2 right there.
Really nice.
Follow you
Thank you,
down with
pilots.
the camera.
All right, doing a rock box,
Starboard in.
please.
Coming in.
I think you still have partial on your main HD as well.
Oh, thanks. Yeah, video can come wide, main HD, please. Sir.
Starboard rock boxes all the way inboard. Okay, you're clear coming outboard with starboard.
Yeah, watch it. I don't know if you saw it, but I got 2. There was a smaller one tagged along for that sample.
Oh, good to know. I'll write
There's
that. I'll record that down.
It's kind
Thank
of
you.
in the shadows, but it's in there.
You got-
Watch lead nav, would you like me to record that as 2 samples as well?
let's just have it as the same sample. We'll record it in the lab as a secondary.
Copy that. Thanks.
All right, arm is off.
Geologist got a 2 for 1.
Okay, blue view set back up. I'm going to go ahead and switch my screen
Thank you so much,
back to
pilots, for
Sirius.
persisting
Yeah, of
on
course.
that one.
All right,
Back up.
ramping down hydraulics, disabling HPU.
Thank you, co-pilot.
Should be good to go. As you come up and out, do you want your swing arms back out? You had your lowers tucked in. I can deploy them back to 45.
Yeah, that works.
All right.
Thank you.
That was a great collection. Thanks so much. Today, on this interesting deeper dive, we took 2 rock samples early in the dive, trying hard to find some of the igneous material that's building up these large boulders and maybe broken from up above to give us clues
Still
about
have you at
how this
35.
seamount was
The ship
formed,
was stopped,
how it
I
fits
believe.
into the geologic
See,
history
this
of the
feature's
New England
10
Seamount
meters out
Chain.
from me.
Since this is a previously
Just
unsampled,
stretch out a
unmapped
little bit to it. See
seamount,
that?
it's a really important data set that we can use to put this seamount into context of the others in this region and the age, which will then give us clues about its connectivity, how is its age related to other seamounts, and how does that affect the life and the geology in this region. So thanks very much for that excellent second rock collection.
This big rock there. I think that's the top. I'm going to rise a little bit, co-pilot.
Coming up as you do.
Just want to make sure I can see over it. Alright, see, I'm starting to see over it right there. Alright, so I can see past it now, up to 40 meters. Looks good.
Oh,
We got this large feature here, seeing lots of fractures on these really, really big
I can see all the way
boulders.
back here.
You
How do you
can
guys feel about
see
15
in
meters?
the
Feel
Same
comfortable with
direction?
that?
Good work.
sonar in the blue view
I'll put it
camera
in.
on feed 3 that
Come
we're
back over to a
looking at
285.
these
Bridge,
really
ROV
hard,
nav.
highly reflective, bright-looking boulders as we go
Ready for another
up
ship move, please?
and over this
15 meters
small
range.
ridge feature.
Bearing 285
So you
degrees
can
at
see the
decimal 2
really
knots.
nice, high reflection off of all of these different boulders as the ROV looks forward to see where it's going. We can see lots of broken igneous formations here, covered in sediments. Definitely a region that's received a lot of mass wasting downslope of material that's been deposited here, and sediments also falling likely from downslope, and also from the water column accumulating into these sediment flows at the base of these big rocky outcrops.
Let's take a peek to the south.
And so, hi Kira, this is Kim. I just have a question. Are we expecting a lot of crusting in this area? Is this kind of something that you would think of a continuation from the corner-rise seamounts as far as geology goes?
Yeah, we're definitely seeing a lot of crusts in this region. Ferromanganese crusts coat the majority of rocks in the deep ocean as iron and manganese oxidizes onto the surface. And so we can see the blackish color to most rocks is often because of that coating. And you get a lot of clues about how long these things have been sitting here based on how the surface of the rock looks, where we have these really angular looking surfaces and not a lot of ferromanganese crust. That means it's probably been deposited here or fallen down into this location more recently, since crusts grow about 1 millimeter per million years. And yeah, in terms of what we're seeing compared to the corner-rise seamounts, they're similar. They're in similar parts of the global ocean, but the new corner-rise seamounts being much further east, we're seeing a lot more sedimentation as we enter the New England Seamount Chain. You'll remember from the past several dives, very coarse-grained sediments and not a lot of buildup. The current regime out there was very strong, especially at the shallower depths. And here already we're starting to see more sedimentation, and we'll probably see that as we keep going west towards the east coast of the U.S.
I don't think we've changed them yet.
And so, yeah, we're seeing a lot of what we expected as we move in. Yeah. And it's also interesting to look at these deeper depths compared to the last several dives. So we're seeing a lot more igneous rock versus the carbonate we were previously seeing. Lots of marine snow floating through here.
Thanks, Kira. Pilot, can we get a snap zoom on some of those corals?
Yeah, up top.
Yeah, no rush. I'm seeing quite a bit of also dead coral stalks. I don't see a full coral rubble debris field, but on this boulder specifically, I see a lot of these leftover stalks that also look that they're encrusted with some ferromanganese coating.
Yeah, we've been having a lot of interesting thoughts, trying to think about why one side of this large boulder has a lot of old dead coral coated in that ferromanganese, and then the other side is where the newer life is still growing. It makes you think about sedimentation rates over time, or perhaps current directions have gotten stronger or changed as to why they are growing on different sides now.
All right, Biddy, let's kind of float here. You want to come on in?
So it looks like I'm not seeing any open polyps on this. Oh, it looks like we have another one of those weird morphologies on a branch that just came out of view. Oh, there's a barnacle. If we move slightly further up, we can see that weird morphology that was displayed where we had associated that with usually an anemone that was kind of surrounding and what we thought likely was forcing then the coral to conform in...
Yeah, if you go down.
A little lower where that weird
right
morphology...
there.
Yes,
Right
thank
there.
you, that. Thanks, Pilot. So, yeah, it looks really interesting to see. Does anyone from the chat room have anything else input? This looks like what we had seen before, but usually with an anemone in the middle, and I'm not seeing an anemone currently here.
Hi, Kim. Yeah, I totally agree with everything you're saying. It does look like there is some evidence of maybe some long ago damage. You can see the skeleton's a little bit thinner in that area. And this is a very old coral. It's very large. You can also see at the base, there's a really big difference in the size of right at the base and then where the first two branch points come out. It's probably because this coral settled on one of these black twigs that we're seeing everywhere, and so that's just an extension of the tissue down over the black twig. But it's an old enough coral that maybe that anemone was there sometime in the past and isn't any longer. The coral's in good shape. It's certainly alive. The polyps have pulled their tentacles in, kind of wrapped them around the top, so you can only see these little red dots where the tentacles would emerge. So it's kind of an interesting pattern that we're looking at. Nice nodal branching. So yeah, very unusual. We have a lot to learn about what it is that causes this odd branching pattern, the circumstances that lead to it, and why you get this sort of cage growth.
Aim at the base,
Yeah, and I noticed that it was... Yeah, definitely. I noticed that there was also nodal branching, not internodal. And do you have any suspicions as to why all of the polyps were drawn in?
No, that's a good question. Presumably something has disturbed them. I don't see anything on the colony. Perhaps a fish swam by. Typically, in my experience from the years that we've been doing these explorations, it's not that it senses the bow wave from the ROV itself, so I don't think that's what would cause them to retract. It's usually some kind of
quiet if you wouldn't mind spraying up your mic.
Yeah, perfect. Is that better?
Yeah, a lot better.
Physical experience. So something has intercepted some of those polyps, and then there's kind of a chain reaction where the polyps next to one another start to close up, and then that spreads throughout the colony.
Scott, is there a reason why the polyps were so
right-hand side
spread out on the colony?
You say it's center now?
Not a good one that I could give you. There's definitely different growth patterns among different species to how dense and clustered the polyps are with respect to one another. So on some species, they're very far apart. Others they're not. But then layered on top of that taxonomic explanation, there are various proximal explanations. How much food there is, how many polyps can the colony sustain. Sometimes maybe it's better to have more polyps when there's low food because all the polyps have a chance to get it. Sometimes it means, well, there's just extra tissue you have to sustain. And so I'll reference back to the earlier comment about the cage-like branches. We have a lot to learn. All of these would require experimental manipulation, designing some experiments in situ, following them through time, and seeing what are the factors. Here it looks like maybe a Venus flytrap anemone is on this large bamboo coral down below.
Yeah, thanks, Scott. The pilots are just starting to set up to get a better view of it. This is quite a large coral. I think, is this one of the largest ones we've seen so far on this dive?
I'm sorry, starboard lower. Can you stow that, please?
Yeah, I would say that it is, actually.
And it's interesting to see that this flytrap anemone can be so high up what looks like on very fragile branches, but still hold up in the water column.
That's great, thank you.
Yeah, so we've noted this a few times. There's a level of buoyancy, of neutral buoyancy that these various organisms have achieved. We are in water. Sometimes it's hard to tell that, because it's so clear. There are so few particulates there. And these organisms, like this anemone, are largely water themselves, and so that's got the same density as the surrounding water. And so it's not like there's a huge weight on the colony. If you pulled that sea anemone out of the water, it would feel heavy to you because it's full of water. But so because it's in water, there isn't all that much difference in that weight.
Well, that's good to know. Yeah, this is quite large. I can't exactly see where the lasers are on this coral, but it looks quite extensive.
You want to come on in?
And it's also, it looks like also, if not all, but most of the polyps are also drawn in. I don't see any out and about, and I wonder if that's why.
Yeah, I agree. Yeah, that's interesting. Second one that we've seen here where the polyps are contracted, so we're not seeing the tentacles. I'm also not, interestingly enough, with these tentacles contracted or pulled over the mouth, I'm also not seeing the sclerites protruding, the needle-like or rod-like sclerites that help us identify these things. This is probably what we would call J-clade bamboo coral. There is a genus of bamboo coral called J. sinensis that is in that clade, and so that's the nearest relative to what we're looking at here. I see there's also a solitary hydroid on the right side of your screen that's growing off of this bamboo coral. All these polyps are kind of contracted like little mounds, and you notice the polyps aren't disappearing into the tissue. That is, the tissue's not thick enough for the polyp to go entirely in it, so it's very easy to see where the polyps are. Whereas when you look at plexaurid sea fans, for example, the branch may appear very smooth because the polyp retracts all the way inside the tissue. That's not the case with these bamboo corals.
Yeah, and this is a great shot of that fly trap anemone that Scott was referring to earlier.
Pulling for full anemone.
We're talking about good questions in the deep sea. And another thing that we don't know is, why is this anemone growing on this coral and we haven't seen it growing on others? And it may be just, the luck of the draw. The larva of that anemone, it'll be moving through the water column when it's at its larval stage and has to intercept this this coral. I don't know how likely it is if that larva had let's say, intercepted a rock next to the coral, how likely it is that that larva could crawl all the way up that colony to then begin to grow there. I mean, it's possible. It's not impossible. But that's, one of the other questions that we would have to identify. So if it does require landing on it at that level, then you could see that the probability that happens is pretty low, which is maybe why we don't see it all that often.
Copy that. All right, video, move on. Nice shots.
Full wide.
Still looking at you, 3-5.
Copy that.
I'll come up as you do. Looks like
Chris
we got
Kelly
a
is
hard
helping
turn.
us out here and pointing out that the genus for
about
this anemone would be
Is that
Actinoscyphia.
40 meters out?
37, yeah.
Like a Chrysogorgia colony
Much
in
lined
the
up.
background there. That
Looks
would be
good.
yet another octocoral in a different family
What status of ship?
of the Octocoralia.
Should be stationary, I believe.
Divide up all the
Been
species.
stationary for about 10 minutes.
Yeah, my swing is done.
You guys ready for another move?
Typically, you hear us describing
real quick
or
to see
discussing
that.
the family names and the genus names. We don't often get down to species names because that would require
See, 10 meters out.
on-board or
Some
in-lab
hard
inspection
returns.
of
I'm going to come up a little
microscopic
bit more,
characters.
co-pilot.
There's
Cover
another
that.
very large bamboo coral colony. So we're getting into an interesting zone where there's either enough stability of the substrate that these
hard items out.
colonies
Do you wanna
can
start
grow
with
to
just
very
10
large
meters?
size,
Yeah, it's fine.
combined with
Closer,
enough
we can explore.
water
Does
flow
285
and
still
food
work?
availability that there's enough
Or
energy
280
to
better?
sustain the amount of tissue
285
growth
looks
to
good.
get to
You're
this
kind
size.
of right there.
Very nice.
285 and 10 meters.
Thanks, Steph.
Yeah, that's a great note, Scott.
We're gonna
We're
bring your lower
about
back up.
2,760
Yes,
meters deep. If you look on live
Great,
stream
Jarvina.
3, we have our quad view, and it's some great
I would like
footage
to
of
request shipment,
D2,
please.
as well as
Range 10
our
meters, bearing
specs
285
for
degrees
both
at
D2
decimal 2
and
knots.
Sirius as far as depth ranging goes and altimetry. And we also have a high-pack screen to help you visualize
You
where
got it,
we
thank you.
are in relation to our current location, as well as a sonar, which is actually a new item that's been put on D2 to help the pilots track where likely they are seeing certain items or coming up on certain objects or features that might we might not see well in advance because light can only travel so far in the ocean. So these sonars are actually really great at detecting, and you can see those. It's in a fan shape, and it has various numbers written on it, and that's actually distance from where this is, so pilots can use that to help coordinate and help maintain the vehicle as well as the safety of the vehicle to make sure that any features that come up, nothing is a surprise.
We were expecting, right? But further up.
Yeah, correct.
What does HIPAC say that slope is here?
And Kira, we're looking,
At
it looks slightly different from what we saw
Yeah, let's
before.
do
I'm not
that.
sure if this is a similar environment that we saw at the beginning of the dive, but it seems a lot more sedimented, as well as a lot of these more
about 60 meters
crumbly,
and about 30 meters rise, so
like
50%
cobble to
slope.
border boulder-sized rocks here that maybe have come up from the top. What is your perspective?
Yeah, I agree. We've sort of left that
should be
really
about 45
massive
degrees or
boulder
so.
field that looked like a really big mass wasting event, and now we've come over to what looks more like a chute of sediment coming down with some ripples. It's been here long enough to have gotten some ripples in the sediment from the currents flowing over and some smaller cobbles. So definitely could be still downslope material that's fallen here and created all this jumble of rocks and cobbles and boulders of different sizes, but not as massive of a large mass wasting event as we were previously looking at with the really large boulders. And definitely you can also see in the ferromanganese encrusting when you get into these more quiescent environments where it's a little bit less active giant mass wasting event, you can see you get these more lobe-like textures of the crust and the rock. You can see here that and by lobed I mean sort of bigger, smoother bumps. That indicates that ferromanganese has been able to precipitate pretty uninterrupted for a long time. And when you have the really bumpy textures, you get a clue that it's still actively precipitating. And so we still have some sediment cover on the top of these features, but the sides and the ledges where the sediment is kept swept free, we can see really nice botryoidal, which is that grape-like texture on the surface of the freshly growing crust. So just, meters away from each other, we can go from where we're looking now at this giant boulder field of a mass wasting event into somewhere a little bit that seems like it's a little bit more settled.
Take a peek, looking north.
Sure looks like more of a 45 degree slope, doesn't it?
Yeah, this side. The other side was pretty. We'll do a quick comparison. So it's to the north. I'll show you south.
Yeah, and it looks like we're heading up a pretty steep slope here as we're moving on up our transect up this cliff feature. We can see, we're starting to see slowly more corals in this area, but I'm still seeing quite a few of those dead stalks around the larger boulders that are there that have been then encrusted by that ferromanganese.
Yeah.
This is an overall general, really great view of
Definitely agree
the
that
entire
the
environment,
slope
and
is
that was what
starting
Kira was
sooner
talking
than
about
HIPAC
how
indicated.
fast we can go from, cobble size to small boulder size stones to like these larger boulders indicating that really large mass wasting event. So great view, thanks pilots. Have we seen any scleractinians yet on this dive? I'm not familiar yet. So far, in my eyes, I've only seen some octocorals.
I feel like I've seen it thrown around in the chat a few times, but I can't quite remember.
Cover that.
Let's see what we can see on the review.
I think we did have a close-up of a cup coral at one point very early on. Rian was very interested in it. But I honestly can't remember if we saw any live tissue on it at this point. But that's the only one I recall.
Out, nice return. Starting to see over it,
Oh, that's
though,
interesting.
so that doesn't look too
Did
bad.
it look like it was encrusted at all, or did it still look like if it was dead, it was freshly
Nav,
dead?
That move end?
About 3 minutes ago.
I wish I could remember. I probably wasn't paying enough attention at the time.
Yeah, it was only a 10-meter move on our previous
no
move
worries.
as well.
I'm sure Rion will catch us up as soon as she
285,
returns.
15 meters.
Looks good.
Copy that. Put it in.
Makes sense.
No problem. Bridge
I
RV
seem to remember a cup coral under the ledge that
Another
had
shipment, please.
at least some fleshy material that
Range
looked alive.
15 meters,
And
bearing 285 degrees,
Tina
at decimal 2
Molokova
knots.
confirming that there was one that was absolutely alive with tentacles.
You got it.
Okay,
So
great.
it's a good thing that she has a good memory.
Yeah, and I
go.
remember,
Sorry,
at
there's
the time when
voices
we
and
zoomed
layers.
in, what caught Rian's attention was that it was pink,
Watch
which
it,
would
you
suggest
hearing me?
that there was live tissue and not just encrusted.
Yeah, we've had the ship moving consistently.
Kim, you had asked
So,
about
yeah,
a
we're just
species that you're
doing our
particularly interested in
normal,
looking for.
track, going back and forth and heading up. Oh, I just, sorry, I understand what you meant. I rose to get a better view of the blue view so I could see further. I just did a quick scan and then came back down. We do that
Seeing here this
periodically
large
just to,
boulder with some
make sure,
the little pokey things
see what's
sticking
ahead,
up for those
get a
at home
size.
watching. Look to be dead coral that's been the majority of it has dropped away and just the stalk is remaining, and it's black looking likely because it's coated in a thin layer of ferromanganese crust that we sometimes call the patina, making it that black color like the rocks around it. And seeing lots of ferromanganese encrusting on these big rocks, creating very loamy texture and some sediment likely cascading from above, sort of like a waterfall of sediment building up on the tops of these outcrops of igneous rock.
Delta 17 there looks good.
Yeah, and some of you may have noticed that the pilots looked like they were coming slightly higher than normal, and that's actually one of our pilots right now was informing me that, you know, it was a great way to get a view perspective of using that sonar that can detect higher up just to make sure that there's nothing unpredictable coming up, so that when they transect up and use the light to actually see, they're not going to run into any surprises. So that's what we talked about before when I had mentioned the blue sonar view. So that was a great example of how they're able to use it in order to predict where they're going to go and to make sure that everything is safe for the vehicle to move forward.
I'm still really impressed to see in this region, you know, for the dives that I was leading for the past 4 or so, the sediment grain size over on corner ice seamounts at the shallower depths at least was very coarse. We saw a lot of really large grains and coral rubble, and here we're seeing the more typical finer grained sediment as we move inshore. And it's just a totally different landscape, having these big igneous flows coated in ferromanganese crusts, and then lots of finer grained sediments in between. Really looking different at these deeper depths and further inshore.
Looks like you have a wall that may have some interesting things on it.
Yeah.
Definitely starting to get steeper. I don't think it's that tall where we came up and looked over. But
so I'm just back from having my lunch, and I hear there was a question about cup corals, and yes, we did see one live cup coral very early on in the dive, almost just after we landed, kind of right under an underhang, had its little tentacles stuck out.
Yeah, it's nice and steep there.
Yeah, if you look at Sirius, it looks like it all comes from one spot and kind of fans out as it fell down.
Yeah.
I was curious, Rianne, as a follow-up question about the cup corals and other scleractinians. I hope I said that right. Do they have a typical depth distribution, and are corals affected differently? I know as we go deeper, the pressures get higher. Just curious, what are the effects of depth on scleractinian Do we know? Are we still exploring that?
We're still exploring it to a certain extent, and yes, you did say that right, scleractinian. So the scleractinians we tend to see usually a little bit shallower. We think that their distribution is affected by kind of a combination of temperature and also the aragonite saturation horizon. So because they precipitate this mostly aragonite for most species, calcium carbonate skeleton, they need a certain amount of calcium carbonate in the water to be able to precipitate that
really
out.
condenses down
And
about
so
16 meters.
we do see species that occur under that, and so we think that those might be more the distribution affected more by things like temperature, but it's kind of a combination of those factors really. The ideal depth band for these deep water scleractinians seems to be about 1,500 and up. Kind of we see a zone in the 1,000s or just in the 900s of where we see a lot of different species occurring. Seems like we've definitely started to come up against the wall here, kind of coming up a scree slope here, and this looks like the bottom of the wall.
Yeah,
excellent.
Contours.
Yeah, and Tina's also just saying that, you know, some scleractinians, as I said, live below that saturation horizon, and we get some Fungiceathus and some Flabellum species that actually live below 5,000, 6,000 meters even, and there are likely other species even further down than that.
It's looking up there.
Okay.
It's nice and steep.
Is that an overhang?
Little one, yeah, right there.
Big coral on the upper left of that one.
Yeah.
Thick base.
It's a good spot to be.
So it's really fun when the ROV takes a look upward to see where we're going. It reminds me of being at the base of a rock climb and sort of looking at what you're getting yourself into as you look up the cliff and see the features you're going to come to. It's a really neat perspective.
Yeah, I really enjoy it too, kind of getting a little glimpse of what we're going to go up against in just a few moments. And you can actually have a pretty good view if you go to live stream number 2, and this is the Sirius cam. So we're the ROV that we dive is a 2-part ROV, so it has the Sirius and then the D2 system. The D2 system is what we're collecting, what we're using as the primary system, and then Sirius is what we call it takes the waves out of the equation for the D2 system. So that part is the part that's directly attached to the vessel. Watch Sleep Pilot.
Hey, watch it go.
It'd be great to have a zoom on some of this fossilized material that's on this boulder here.
Yeah, of course.
Thank you. Just as we go by.
Let's see. Hey, video, how about right there? Is that good? You want to come on in? Dark background. Does that work?
Yeah. I was wondering if we were starting to get into the zone.
I just want to snap. I can always get
of seeing these fossilized cup corals, but I don't think we have. I think this is still mainly the bases, potentially octocorals, different kinds of corals. You can see that covered over in that ferromanganese patina. There's a brown one there and a black. And so these are pretty old. These have been here for a very long time.
Is that all you needed, Watchley?
Yes, that's great. Thank you so much. Beautiful Brisingid and a Stauropathes in the background there as well. That Brisingid with his arms really out wide. You can see that sea star in the background there. Beautiful.
We'll do some side-to-side views.
Another Chrysogorgia. We've seen a couple of those so far.
To the north.
Fish in the Sirius view.
Video, did you say something?
You're saying there was a fish in the Sirius view?
Oh, copy that.
Co-pilot.
Co-pilot, can we do a zoom on Sirius?
Sure.
So we're just going through a pilot watch change right now, and then we will continue with our dive in a moment.
Pulling Sirius out.
Thank you, video. Okay. Seems like large boulders before the cliff.
Bridge ROV nav. I'd just like to confirm that the ship is stopped. Thank you.
Looks like my heading's 285-ish. Looks like
Yeah.
you get your sonar looking a little bit up, 11 degrees. And pretty much centered.
All right. So we just did a shift change. Watch lead, Josh piloting, Levi, co-pilot, and Jim in nav.
Perfect. Thank you so much.
And Roland on video, of course.
This large Persingid sitting atop the rock here with his arms hanging out. On one of these bamboo corals. If you could have a snap zoom on this bamboo, please.
The large one?
Yeah, the really big one.
Yeah. Come on in,
Thank
video.
you.
Let's do a partial and I'll swing my heading around.
Oh, this one is still alive. Okay, so we've seen a fair number of skeletons without any polyps on it. But this one does seem to have polyps all closed up. Lots of barnacles as well sitting in this particular coral.
Rian, while you were out on lunch, we had a really good view of a couple of these colonies that were in good shape, like this one.
Yeah, you can come on in.
Do we know what species these might be, Scott?
On them, though. If you were going to ask me if we know what they are, I suggest it's J. clade, which would be currently Jasonisis. And the interesting thing, we were making this observation before, that in both of the colonies that we stopped to have a look at, the polyps were just like what we're seeing now. All the tentacles were pulled in. And so you know, at the first one we saw, when Kim asked me about it, I suggested something had impacted the colony, a fish or something that caused them maybe to retract. But I'm starting to wonder if perhaps it's a time feeding thing. You know, the tentacles would be extended for feeding, and maybe they have a certain time frame, or it's when the current is bringing enough food. Maybe there's just not enough food drifting by right now to bother putting the tentacles out.
Right. I did see there was a branch on this colony that does have its tentacles out, but just a single little area, not a big area. Nice shot of those barnacles there.
See if we can find that open polyp.
So these barnacles are crustaceans, so they are related or in the same phylum as things like crabs and these spot lobsters we often see in the corals, but they're fixed. And barnacles actually are sitting on their head. It's actually their legs that are sticking out the back end. And that's the feeding apparatus that you can see. So there we go. There are some of these, some polyps that are out there, Scott. Just one single little branch there.
So it's so interesting, you know. I don't know why these ones are out and all the other ones are tucked in, and the previous colonies we saw were tucked in.
It's really curious.
One thing that is informative is look how different the polyps look when they're extended with their tentacles, and how much they can contract and pull those tentacles down. And what they're doing is, it's imagine your fingers in your hand, and you curl them in toward the palm of your hand. So don't just lay them flat, but
Dan,
you curl them
the
inwards
jar
so
camera
you don't see the tips.
And
you only see that little
the
red
monitor that
opening,
has
so
the
that would be
Thanks.
basically it's an opening among all the tentacles, and underneath that is where the mouth is. And then they can contract some, but they can't contract all the way into the tissue that's on the branch. So you get these little sort of volcanic-like mounds go along the branch.
Yeah, it's curious.
Very curious. I was going to say about the barnacles, you know, one of the earlier colonies we looked at had this very large Venus flytrap anemone on it. And I was noting that you had to be lucky, or the larva of the anemone had to be lucky to impact that colony in order to grow in that spot. And it's the same with these
Bridge ROV
barnacles, you know, as larval stage.
I'd like to request a move, range 10 meters, bearing 285 degrees,
where they want
speed
to
decimal 2
and grow
knots.
as the adult stages that we've just seen there. And so there is some element of luck that the current carries them to where they can detect that colony and orient to it. And so, you know, that's one of the things that's controlling when we see these associates that are attached to the sponges, to the corals. You know, it's different for something that's mobile, like Brasingid or a feather star, crinoid, or
I don't think we added weight.
sea stars.
How heavy was it?
So on.
Yeah, absolutely, Scott. I would actually guess that it's actually a lot of luck for those larvae to be able to find a suitable location to settle.
About like 20 pounds?
I mean, so water is, what, 62 pounds per cubic foot?
Yeah, but it's 10 pounds, 15 pounds. In water? Oh.
It's an interesting little look around the edge of this wall. So we started this dive in a kind of sloped area that had a lot of sediment ripples, and now we have moved our way up to a wall feature. And one of the goals of this dive is to make it up this wall feature and onto a plateau, exploring and seeing what geological features are here, and also what the biology composition is like on this wall area. So far, we've definitely seen a handful of deep-sea coral species and a handful of deep-sea sponge species, as well as some Brasingids and sea stars and a few fish. We are currently at 2,730 meters depth. There's a lot of sediment on this slope, this rise area here, Kira.
Yeah, absolutely. You caught me just coming back from my little break, and yeah, it's really fun to see it pooling up on the upper surfaces, giving us a clue. Sediments in the deep ocean are always really interesting because they hold a lot of history inside of them. What kinds of organisms have died and fallen and accumulated in that sediment? How much organic matter is there, and what can that tell us about productivity in the surface waters? What kinds of minerals have built up there over time as they've fallen through the water column? So even though it just might look like dirt, just like on land, sediments are actually really interesting, and some scientists study them specifically. So regions like this, when we see a lot of sediment cover, it gives us clues about what's been going on in this seamount over its long geologic history. And some of these are so sedimented, it's almost looking like a different rock type, but I think that's just sediment filling in every little nook and cranny of the ferromanganese coating, creating sort of almost like a carbonate-like texture. Very interesting.
Yeah, it's really interesting. It's interesting to see quite this much sediment, too. I feel like on our previous dives, we haven't seen quite this much on these kind of slopes. It seems like a lot.
Yeah, I think part of that is because we're a lot deeper, so we're at a depth where there's an opportunity for things to flow down slope and create this mass wasting, which is what we call when debris flows down slope. And so we're in a bit deeper environment, which gives us a different regime. And then also I think the sedimentation rate, just as we move inshore, is going to increase and also cause increased rates of sedimentation. So I totally agree. It's a bit different because I think we're further inshore and we're also a bit deeper.
Yeah, that's a really
fun to see how those different environments.
Yeah, it's a really good point that we're further inshore. So right now we're sitting at about 840 miles into Newport, Rhode Island, where we came out of and we're going back into. And of course, when we were on the Corner Rise, we were closer to 1,300 miles away from Newport, Rhode Island. So we really have come inshore quite significantly, even though we're still at the kind of far eastern end of those New England seamounts. It's like some more of these large bamboo coral colonies. I can see some smaller sponges as well in the area. Lots of particles in the water. So these particles in the water are what we call marine snow. And marine snow is really a mix of things that have died in the water column and a little bit of sediments and other material. And this marine snow is really what feeds the deep ocean. So these seamounts in particular interrupt the flow of cold, nutrient-rich deep water. This deep water flow hits the bottom of the seamount and kind of curls around the seamount and coming to the often very surface waters. And in the surface waters, all those extra nutrients can cause plankton blooms. And as those plankton blooms die, that material comes down into the deep ocean. So the organisms here that live in the deep ocean, they don't photosynthesize, they don't use sunlight directly, but they are feeding off organisms that have used sunlight during their life cycle. There's a question from Harold Carson in the scientific chat room about so do deep sea corals tend to have large dispersal potentials in planktonic larvae durations? And the question to that, I'm afraid, Harold, is we really just don't know. We know just a handful of species. We know about the larval biology of just a small handful of species, but what we do know so far is it can be very species specific. Some seem to have very dispersive larvae that can travel. We're seeing pelagic larval durations of several months to even up to a year. And then other species, produce a crawling larvae, but the larvae really don't go very far and so remain fairly close to the parent and settle and grow. And certainly we see that there are certain species in the deep ocean that we find in almost every ocean. And so those species, just by looking at that biogeography, you can say, okay, that species really must have a dispersive larvae to be able to have got so far. And then we see other species that are very specific to relatively small areas, small chains of seamounts, or even just continental shelves. And those you can hypothesize that they probably don't have as dispersive a larvae as others. Beautiful shot of the polyps there on this bamboo. So these polyps are out, Scott, if you're still on the line. There's a couple that are in there, but most of these are actually out. And it's beautiful.
I am still here and watching and typing. Very multitasking, these dives, aren't they?
They are very much. And so what Scott's talking about is we have in the background.
In a lot of different ways. Because all the scientists are communicating by chat in the chat room. We're also typing observations when we should be watching and talking. So yeah, what's the difference here? Why are all these polyps out and they weren't in the last one? Ken Sulak made the interesting observation that on that last colony we were looking at, it had the barnacles, and the barnacles were feeding. All of these things are suspension feeders, grabbing things out of the water column. So there was something there to feed on. One thing that's different about the barnacles and the bamboo corals is just kind of interesting to know is that the barnacles are active suspension feeders. Okay. That is, Rian referred to them growing on their heads, and that you were seeing the legs sticking out of the shell.
Turn a little bit,
and those legs actively sort of pump through the water column. They're kind of reaching out and fishing through the water, whereas the coral polyps are passive suspension
Bridge
feeders.
ROV
They
nav.
just sit, and they really have to rely
I'd like to
on
request
the water
ship
passing
move,
over
range
them.
10
and then
meters,
if something intercepts
bearing
one of those
285
tentacles, then
degrees,
perhaps they can
speed
grab it and
decimal
feed
2
on it.
knots.
And so that may be a clue. Only when there's enough current flow for the passive suspension feeders to have their
good copy, thank you.
Tentacles out. Maybe that's one reason. Boy, that's a really big colony, isn't it?
That's a really big colony, yes, and there was another one behind it there as well, and several. It looks like there are several upslope here as well. Really interesting.
Your mic's open, Roland.
Sometimes you have to take a breath and just enjoy what you're seeing.
I do that very often. It's really, for me, it's really a joy to be back here in the back row and being able to watch these ROV dives live and see these areas of the seafloor that have never been seen before. It's really really just beautiful and stunning. Very, very blocky texture here. This is really, really interesting. When we climbed up some walls in the corner rise, we saw these large, potentially carbonate walls with all these holes and texture in it. But overall, as we backed out, they looked relatively smooth, whereas this wall is very, almost crumbly and crusty. A very, very different morphology, at least from my untrained biological eyes.
Yeah, I agree. I definitely think we're seeing more igneous morphology where you can imagine the rocks cooling from their magma state over time and creating these big, large bumps and crevices, and then getting an additional rugosity from the ferromanganese crust that we're precipitating on the surface of that, creating that more micro, or smaller scale texture to create even more bumps. I was actually going to ask a question to Scott and Rian and anybody else who knows as we zoom in on this coral, I'm sorry, not coral, whatever it is.
No, the corals, two little coralliums. two coralliums that we're seeing here. They are coral. Yep. Go ahead, Kira.
My husband had been tuning into the video recently, and he was curious, have we ever tried to use any different light spectrums to look at deep-sea organisms? On the ROV, I assume it's visible light. That's why we're seeing it, and we're interpreting the colors as we would see them on shore with, light from the sun. But just curious if we've ever tried looking at them through any other light spectra.
Copy that. Thank you.
That is a really good
wavelength.
Yeah, that's a really, really good question. I know I haven't personally, but there have been expeditions that have looked at a lot of these corals actually bioluminesce. And so they've gone down and turned a lot of the lights off to look at the bioluminescent patterns. I don't know if Scott knows any more about whether they've looked through other spectra than that. But
yeah, Ian, I think that would basically be my answer. On occasion, not very often, but on occasion, D2, we've turned off the lights to see if we could see any evidence of bioluminescence, but we haven't done that filtering. I believe on those bioluminescence-dedicated research expeditions, they have been doing some stuff like that. It's kind of an interesting question. You know, it gets back to, well, what are the wavelengths of light that are even available down here? I suspect, you know, a lot of the color that we're seeing isn't really related to the visible spectrum that we're seeing. You know, we bring down the lights, so we see all this color. The color is a secondary byproduct of something else that's going on. You know, it's usually dark down here, and so you couldn't really use the color. I don't know what sort of special eye structures would be required to I mean, I understand you would need certain pigments to be able to see different parts of the spectrum, and some fish certainly have those somewhat shallower than we are now, but I don't know down at this depth if that would be meaningful.
Recommended.
Thanks so much for those great answers. I'll be sure to pass it along.
Yeah, the bioluminescent patterns in the corals are really quite beautiful. The bamboo corals in particular tend to bioluminesce. Here we are zooming in on a sea star. Sorry, go ahead, Scott.
Sorry, we're going to do the sea star, but we can get back to it later. I was just going to say, I think you recall, didn't we look at some of these bamboo corals in the cold room on a? We did. The bioluminescent flashing along a branch, but yeah, let's look at this. I think this is a slime star. It's really interesting. All of these spination patterns you can see.
It's a really interesting.
Like that little pocket at the top that suggests it's a slime star.
Yeah, the sea star is almost fuzzy with all these little spines. It looks like a little fuzzy bear. Beautiful.
I think that this is probably one of the best examples I have of why this phylum is called the Echinodermata, the spiny skin. This is great.
And your port upper.
There we go. Thank you.
Yeah.
Lovely.
Beautiful.
Is that its mouth? What part of the sea star just opened up?
Here's Chris to tell you all about it.
Yeah, Chris, great timing.
The mouth is on the other side.
Yeah, we have some questions about the operculum there.
In. Yes, well, that was actually what I was going to first explain, was that usually when people see these, the first thing they wonder is what we're seeing about what that hole is on the top. And so this is a pterasterid, or something closely related to it. To be honest, I'm not sure this is hymenaster or even pteraster. There's a bunch of other weird things that live in that sort of general part of the starfish tree that this could be. I mean, I would tentatively assign it pteraster just to sort of put it somewhere, but there's a couple of other weird things. But a lot of them are characterized by having what this thing called an osculum, which is the same kind of word they use for sponges. It's an opening that's on the top of this secondary, what's called the supradorsal membrane, that is present on the surface. So sea stars and the pterasteridae like this, what you're seeing is not actually the surface of the animal. You're seeing this tent-like covering that covers over the actual surface of the animal. And so it requires this secondary opening, the osculum, to allow water to sort of breathe in and then breathe out. The gills for the animal are actually inside. This could be something that one of the Russians described called amembranaster, which occurs quite deeply and has only been seen literally a couple of times. So this could well be something extremely rare. It's one of those things that one of these days I need to sit down and work out all that stuff and figure out which ones are which, and maybe start collecting more of these to figure out, just how many of these are present in these deep sea habitats. But yeah, I mean, it's possible this one might be a full boron slime star, or it could be, you know, there might be a couple that don't. I'm not sure yet, but there's definitely a lot of these really the animals, the sea stars in this group of in this group of the Pterastridae are fairly rare, or rarely seen. So they're always very interesting. Oh, the sea cucumber. I think this falls into, I want to say, that the group that we call the Elpididae, I think, a sea pig.
A sea pig, yeah.
I mean, I might be a little off. I'm not as up on my sea cucumbers as others, but they're the ones that have those weird extended tube feet tentacle things that come off the back, and they have that sort of unearthly, oh no, look at that, looking straight down onto the mouth.
Yeah, it's really interesting.
Well,
I don't think I've seen I don't think I've seen a sea cucumber do this. It's kind of half hanging off the wall and into the water column.
I mean, it could. I hate to say it, but I'm not sure if this is the mouth or the anus.
Right.
I assume it's the mouth. Believe it or not, well, because one of the things that's been sort of learned lately about the shallow water species is that sea cucumbers actually have a respiratory chamber coming off of their colon. So they can actually breathe through their ass as well as other places. So this could well be, I think it's the mouth. I think it's probably just feeding, but it could well be it's holding up its butt into the water to breathe.
Yeah, it's so hard to know which end is which on a sea cucumber sometimes. It does seem to have more of the pseudopodia sticking out this end, so maybe.
Yeah, I mean, 50-50, I'd say 60-40, it's the mouth, though. So,
yep.
Yeah, I think there's some feeding arms there.
Feeding tentacles.
Wow,
yep.
That's a beauty.
Yeah, really unusual to see it kind of just hanging out like this. Actually, I think it's just starting to open its mouth right now.
Oh, yeah. How exciting. Okay. No, I mean, sea cucumbers are so... I mean, we run across them so often, but then there's so many stories of what sea cucumbers do, and they vary across I mean, some of these sea pigs, the Elpididae, some of these actually have parasitic snails that are embedded in their tissue.
Oh, interesting.
And I mean, I mean, there's actually a few. Those snails get everywhere because even some of the what was it? There was there's a couple of urchins, I think, that have. I mean, the deep water ones that have parasitic snails in them as well. And in fact, the other day I was thinking, oh wait, I'm not thinking of. Yeah, that's right. There's a bunch of stuff like that. Anyway, it looks like the echinoderm segment of our show. So, I will let you get back to corals, and great job, and I'll talk to you guys later.
Thanks so much, Chris. Appreciate you dropping in.
Always happy to do it. Bye-bye.
Beautiful sponge right ahead of us. If you see those two red dots, those are 10 centimeters apart. And so this was actually quite a large sponge here.
You're partial.
Yeah. Kind of sitting on this ledge. So these sponges are filter feeders, so they have pores all over their outside that bring in water. They have specialized little cells with little flagella that wiggle, and that wiggling creates water currents that come through the little holes. And as the water shoots through, there are other cells that pick out particles and nutrients and all the things the sponge needs to live and grow, and the water comes out on the other side. And these sponges often form habitat for other organisms too, and I think we can kind of see some of those in this picture here, sitting in all these lobes.
Yeah, it's coming up.
Beautiful. Thank you.
I also love on those zooms when you can see the sediment is just perfect in this region to fill in between all of the botroids on the surface of the crust. As you can see all these really other bumps on the finer scale. Some sometimes when there's not a lot of sediment cover, it's harder to see that texture, but because the sediment is just fine enough and distributed well, it's really sort of highlighting that botryoidal surface of the ferromanganese crust, sort of outlining it in a little white bit of sediment.
Yeah.
Super cool. And we see some erosive features here, these downward cascading channels. On a previous dive, we were seeing this in carbonate, but you can see it happening here in the ferromanganese crust. And Jason Chater had explained to us that it's when sediment gets mixed together with seawater and it gets a bit denser and it starts to flow over the tops of these ledges and creates a sort of abrades away the material and creates these vertical channels. Nice to see that continuity of textures and processes happening between the two seamounts.
Sure.
And the upper portion of the shoulder here, the ferromanganese crust just being actually eroded away. It's really interesting when we sample ferromanganese crust, we can study the seawater chemistry, and when we lose layers, we lose some of that seawater history. Zooming in now on something interesting here.
Yeah, I think it's a little sponge. I can just see the top hole there on the left-hand side. This is where the water gets exhaled. And this little beautiful snail sitting on top of the sponge. These really tight whorls. Really beautiful. Seems very precariously perched on this little ledge on this sponge. And then on the left, the stalk that's going up, you can actually see these are zoanthids. They're a colonal type of, similar to anemones. And they often take over these old coral skeletons. That's a really good zoom there. You can see this beautiful pattern on the shell. Really pretty. And you can see those holes in the texture of the sponge. So the water usually will go in those smaller holes, and all the nutrients and food is stripped out, and then the exhalant water comes out of the large holes. The sponge certainly has a little bit of a coating of sediment on top too. Very nice. Thank you so much, pilots.
Copy.
From the bottom of it.
See these zoanthids up this this old stalk. Beautiful chrysogorgia. As we come up on this ledge here, these ones have those large squat lobsters living in the middle. You can just see that squat lobster's long arms kind of in the middle of this coral colony here. Pretty.
Do a partial on this.
I think we can get another 5 meters in, Jim.
I think Scott France had made the observation earlier on during this expedition that these squat lobsters that live in the center of these corals often they're often females with eggs.
Bearing 285 degrees, speed .2 knots.
Yeah, I think, Rian, when I said that, I was referring to the shrimp. It may also be true of the squat lobsters. The squat lobsters, at least in this family, I believe this is the family Chirostylidae, that unusual name refers to the very long pincer arms. They're like needle arms, which is what that translates to. And so most of the species in that family that I'm familiar with are very host-specific. And so a particular species of this crab will live only with a particular species of coral. And so that would suggest you should find both a male and a female at various times of their life in these colonies, because they don't live anywhere else. You know, you never see this Chirostylic crab just on a rock, for example. So really interesting.
It is very curious.
Hoping maybe later. Yeah, go ahead.
I was just saying it is very curious that there's only one, because we do often see two, a potential mating pair within these.
Right, exactly. So, you know, there's a whole interesting range of questions in biology there as well, right? And so the larva of the chirostylid has to find that chrysogorgia, and then, you know, whichever one lands first, let's say it's the female, then a male has to find it later on in order for the species to propagate. So just all kinds of fascinating biology and sensory biology about finding the host and then finding each other. And hopefully later, you know, we'll find some of these chrysogorgia that have the shrimp in them. And I have noticed on some of the past dives, you can see part of the shrimp is really deep purple. And those are either, if it's in the abdomen, or excuse me, actually it would be up in the thorax. I guess that would be the developing gametes. But I think more often what we see is the embryos being held on the swimming legs under the abdomen. And so there would be this very distinct purple color that you can see pretty easily.
Can I use your panning tilt?
Sure.
Just going to come up. It's like a slopey debris field.
It's like a little ledge there.
Yeah. Or a big rock.
I think we're definitely still seeing here a lot of downslope material that's been transported here. If you look at HIPAC, our navigation map on feed 3, you can see that we're still at the base of this really steep wall. About where the ship's nose is, you can see the contour lines getting closer together, indicating that that region is going to be steeper. And where at the back of the ship shape, where you see our white ROV, we're still at where the contour lines are a bit further apart, indicating that we're at the down slope part of that really steep cliff. So I think that's why we're still seeing a lot of sediments that seem like they've cascaded here from above, and a lot of rock debris piling up in these cobbles and boulders. And so it'll be very interesting to see how things change as we approach this much steeper slope along our dive track.
Yeah, and if you've just joined us here today, I'd like to say welcome to Dive 12 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. And the stepping stone that we're looking at today is a little small, unmapped, previously unmapped and unexplored seamount that we've nicknamed the Y Seamount because of a feature on its very top. We started the dive today at 2,830 meters. Right now we're at 2,701 meters. So we've come up just a little bit over 100 meters so far. The objectives of this dive, we started in a small plateau and kind of worked our way up a really large boulder field, and then now kind of on a high slope area, coming up towards another plateau eventually.
Can you guys tell us something about the geologic nature that has caused these seamounts to be here in the middle of the Atlantic?
Yeah, that's a great question for Kira. Do you want to take that one?
Yeah, sure. Great question. You'll notice if you look at any of the overview maps of where we've been diving on this New England and Corner Seamount Chain, and you'll notice that they look very linear. Like each little mountain is sort of progressing along in a line. And usually that's indicative of hotspot volcanism, where you have a hotspot of magma in one location, and then as the plate moves due to plate tectonics, then the plate will be moving in one direction over the hotspot, and it'll create a volcano, and it'll create a seamount. And sometimes these can even be big enough to be above the sea surface, and then they cool and they sink and create these underwater mountains, and the plate continues to move, and then the magma comes up in a separate, right behind it as the plate moves away, and we have the next seamount in the line forming. And so that's how over time we've created these large chains. And I believe that there are two different hotspots that are thought to have created the Corner Rise and New England seamounts. And so that's part of the reason why we are so excited to sample the igneous geology here, to be able to figure out which seamounts come from which hotspot chain. So depending on where that hotspot is, the element composition, the trace metals, and some of the isotopes can change, and so you can grind up the rocks that we get, especially the igneous rocks, and get clues about which seamounts are part of the same chains and their ages and how long ago they were formed. And so those are some of the processes those are some of the processes that have created this long chain of seamounts and the underwater mountains that we're seeing here today. And every rock we collect helps us put together that story a little bit more clearly.
And from the biological view, these chains of seamounts are really, really fascinating. So a single seamount, as we've talked about a bit before, can really concentrate these deep nutrient-rich waters and creates what we call hotspots of biodiversity. So they have the ability to potentially aggregate life because they're areas that are full of food and really good areas to live. But when you have these chains of seamounts, we potentially see what we call stepping stones. And so that's the ability of larvae from one seamount to travel to another one and potentially cross even whole ocean basins this way. And so they might be conduits for larvae to travel from one side of the ocean to the other. And that might be one of the reasons that we often see similar species on the European continental shelf and on the U.S. continental shelf. And so that's part of our exploration cruise, is looking at these stepping stones and seeing what species live in these different and newly discovered areas.
I love that somehow we both managed to fit the word hotspot into our definitions. Hotspot volcanism and hotspots of biology.
Exactly, hotspots of biodiversity. These seamounts are just hotspots all around.
And definitely what we're seeing in the view is a debris field. You can see some of the downslope tracks and sediment showing where it's cascaded down. Kevin Conrad in the chat, a geologist, telling us it looks like this might be a slump scarp, which is a bit of a tongue twister, but slumping meaning the failure of the slope above, creating this sort of debris field with lots of individual rock pieces that have been broken up as things cascaded downslope, and a bunch of sediment that's probably traveled with it as well as accumulated here. On feed 3, we've just pulled up a nice map of the symmetry here. This is a slope map if you're looking on feed 3, and so the regions where the color is more red, that indicates a steeper slope. And so we can see where we are in our dive, and how, this could definitely be a slump scarp as we approach this more steep face.
Yeah, so on this map...
It's just amazing what those modern maps can show. Sorry, go ahead, Rand, you go ahead.
I was just going to point out that on this map you can actually see the vehicles too. So you can see kind of our track line, which is this lighter line coming up. So we started in an area, kind of that blue-green, which is less steep, and then we've moved up into this red that's steeper. And then you can just see the two vehicles there, D2 and Sirius. D2 is kind of the whiter color, I believe, and then Sirius is the darker color on there.
Looks like I'm moving in. This target's now right at the 25 ring.
Yeah, that's that little mini cliff.
Spire kind of thing.
Yeah.
Yeah.
So you want to wait until you're fully settled out, and then we can bump a little closer?
Yeah.
Fish shadow.
Yeah, I was just going to add quickly, after we checked out that very cool slope map, and we're looking now at the high pack view. It's, here comes a fish. I think he'll steal the show for my comments about that.
Yeah. Oh. This looks very cusk eel-like. Oh. You can actually see in the background there too, there are some of these fossil corals. These Desmophyllum dianthus were just in that area.
Those are huge.
They are enormous. Yeah, some of these Desmophyllum can be really, really large. So they're at least 5 centimeters across.
Oh, I've never seen that. That's amazing.
Yeah.
Yeah, Jim, you could probably bring me in 5 meters. That's not good. 285.
It's potentially a Bassozetus species of fish that we're seeing here.
Bridge ROV nav.
Do one more tight on the fish.
That would be great. Yeah, just a good on the head end.
Range
it helps
5
that description.
meters, bearing 285 degrees, speed decimal 2 knots. Good copy, thank you.
See, swimming around all of that coral rubble, all that, those corals are covered over in these ferromanganese patina, and so have been dead for quite a long time.
Even though it's so pitch black down here, I still notice a lot of these fish have, normal, what looks like eyes.
Yeah, there's really 2 strategies in the deep ocean, Roland, in terms of eyes. You either have relatively large ones to try and get any glimmers of light or bioluminescence that you can, or you have none at all because you don't really need them. And so these different fish have different strategies, different organisms have different strategies in the deep ocean on how to deal with the darkness that we see down here. It's interesting, we're really in an area with a lot of coral rubble, and I know from previous dives where we've seen this, this suggests that we have quite a high slope, potentially a real cliff ahead where all of these fossil coral pieces, these coral pieces have fallen from, and that certainly looks like what we have right now looking ahead.
Josh, is that an overhang? Because I notice there's no sediment on this side of it.
Copy, thank you.
Some sponges on the wall here.
Yeah, it's hard to tell. I think there might be a little overhang, sort of like a rounded overhang to it. Let's get one of these sponges.
That would be great, thank you. Little spiky sponge here sitting on the wall. There are several of these that we could see in that picture, and right below it is one of these fossilized coral pieces that we've just kind of started to see in this dive, I want to say, in the last 20 meters of elevation or so.
Follow your zoom, follow your focus.
You can really see that ferromanganese coating, all those bubbles and botryoidal, and of course all over the coral pieces that are there. So sponges have something called spicules, and those are the sharp points that you can see sticking out. There's actually some organisms on those spicules as well. I see some foraminifera on them. And those spicules give the sponge some structure and also a little bit of protection too.
Yeah, I can't get any closer. Still stretched out.
Yeah, no, thank you so much. Christopher Kelly was just reminding us this is, we're not even sure what this sponge is, so we collected a very similar one a few dives ago in the hopes of being able to identify it.
Okay, let's come on out.
There's some little amphipods in the top there. Beautiful, thank you.
Had a little 5 meter move in.
Really large sponges there in the background too.
Ever so slightly swinging towards you. Keep the arms out?
Yeah, I think we're all okay for right now.
Okay.
I don't know how tall this thing is.
Okay.
I'll just keep perpendicular to it.
Okay. Sounds good.
I have more of these spiky little sponges.
You want me to turn towards you a little bit? Or
Yeah, that'd be good.
Okay.
And these sponges like to have a bit of water flow. They are filter feeders, so need the water to come through and provide their food and nutrients. It's been noted that sponges need usually less of a current than we usually see the deep water corals in. They can survive in a little bit less of a current than corals can.
Is that because they produce their own sort of suction?
They do, yeah. So these the sponges have their they're called choanocytes. They're little specialized cells with little wiggly flagella in them, and those wiggles create a water current, so they can kind of create their own water current. Whereas corals are very, very passive. They have to wait for the water to come by with that food, and so they usually like areas with a little bit more water current. So there's a lot of overlap in where these corals and sponges can live. Could we zoom in on this coral just coming into view now with the forks? Mm-hmm. Thank you.
Okay, let's come on in, roll.
Again, the polyps are all closed up in this one again. We've seen quite a few corals today with their polyps closed. It's like a bamboo of some kind. With a few little tentacles sticking out of one of the polyps. Thank you, pilots. That's great.
Yeah, I'm sitting perfect, Josh. Looking right at it. It's just in my 25 and holding.
Yeah, it's pretty tall, huh?
Yeah, it's definitely tall.
Yeah, I'll pull off and you can still my
okay.
Still my uppers.
If you go over to live stream number 2 right now, you can see the view from the Sirius camera. So this ROV system is in 2 parts. We have Sirius, which is the upper part attached directly to the ship. And so that helps to take some of the waves and the heave out of the wire. And then down below is the D2, which is the main body of this ROV system and what we are primarily looking out of and what we can collect with. But it's really nice having these cameras on top of the Sirius and looking down onto the D2 because you can really get a good sense of scale. Sometimes it's very hard to get a sense of scale just looking out in the same plane.
Look at that little
ledge. Look at Sirius right now. You can really see that ledge and this rock kind of towering above us here. This rock face is absolutely covered in sponges. Lots of these little spiky sponges. There's a Bathypathes coral there.
Coming up a little bit.
And then lots of these fossil coral rubble as well.
Yeah, it definitely seems like we've gotten out of the debris field that's below the steeper cliff we're going up, or ridge, I guess. So we should start to see a bit different morphology as we continue on our dive track. More big rock outcrops and a little bit less cobbles and sediment jumbled below the slope. You'd imagine sort of like the peak of a mountain above the scree field below.
Yeah, absolutely. And as we start to climb this face too, we already really have started to see a little bit of a different fauna on here as well as these fossilized corals that we didn't see earlier in our dive track, and they're really, I can see them all over the walls now at this point. So there's a little starfish here, sea star. You can see those really elongated tube feet. So these sea stars have something called a water vascular system. So they have a little hole in their usually upper surface called a madreporite, and that helps to control water in and water out. And they use water to flow and move their tube feet and create suction on the bottom of them. I'm waiting for Chris Ma in the scientific chat room to let me know on a potential genus or species or even group for this one. Beautiful. Actually, you can kind of just see that madreporite on the surface. It's kind of a lighter yellow circle. At least I believe that's the madreporite. Usually they're relatively obvious. Evopsoma. Yeah. Yeah, look at those 2 feet. Beautiful.
Let's come on out.
And these sea stars are generally predators, so they will be roaming along the surface of the rocks, looking for organisms that are living on these rocks to be able to eat. We see a lot of sea stars are also predators on corals, and they will eat all the polyps off of corals as well. The Chrysogorgia in amongst a field of these spiky sponges. Yeah, so Christopher Marr has confirmed that particular one is often a coral predator, and so maybe it's roaming around looking for its next coral. This large sponge. Yeah, lots of these old and very old and broken coral stalks left out of this this cliff here, along with these fossilized cup corals that are on there.
I think there's a large colony above you, kind of. See that?
We've had some questions about how old some of these fossilized corals that we're seeing are. And we know from previous dating work that some of them have dated back, 10 and 20,000 years. And some of the oldest that have been dated from this region on the New England and Cornwall seamounts have even come back in the 200,000-year-old old range. That's pretty big. Yeah. That's
prime real estate, apparently.
Yeah, we'd love some lasers on that if they're available. So we use scaling lasers to help us identify how large things are on the seafloor, and you can just kind of see through the branches a little bit. 2 little red dots. These are harmless, and they are 10 centimeters apart. And so this coral it is at least 1.5 meters, if not bigger than that across. This is huge. Beautiful.
So 1.5 meters, so 150 years old, maybe? Like that.
Yeah, maybe older. It'd be great to have a snap zoom on the polyps and see if we can get an ID on this one too, if that's possible.
Yeah.
And look at that base. That base is so thick.
Copy. I'm just setting up here.
Yeah, thank you. This kind of looks a bit like a primnoid octocoral.
Go ahead and roll.
Beautiful golden skeleton there. Oh, maybe not. Maybe this is a bamboo. Yeah, no, I'm sorry. This is a bamboo coral. I can see very clearly a node right in front of me.
Yeah, I think this is that same one. We've now zoomed in on several times. They've been building these really large colonies. But I agree with you, Ian. As we were coming in, I was a bit fooled myself.
Yeah, those nodes are really, really conspicuous. Like they're very, very small for such a large, thick skeleton, and very potentially grown over. Beautiful.
Right, and so I think one of the things that's going on here that supports my earlier idea that this isn't a J-clade related to Jasonisis is that taxon has very small sclerites that are shaped like little scales, and they're embedded all throughout the tissue. And so when the light hits them, they become kind of opaque, kind of milky, and so it's kind of hard to see through it. And so that's why when we approached, it looked really white and smooth, and you couldn't see the nodes. And I think it's because of those small scales.
It did indeed.
Unlike several of the earlier ones, most of the polyps are out, extended on this one. And I'm looking closely, and one of the things that's really interesting, I do not see any needles or rods up on the body of the polyp itself or sticking out, protruding between the tentacles. Very fascinating.
No, I don't see those either. Yeah.
Characteristic for taxonomy. Yep.
Yeah, really fascinating. So these bamboos are octocorals, so you've noticed all these individual little polyps. They have those 8 tentacles, which kind of gives it its name, octocoral. And each of these little polyps that's sticking out from this skeleton are actually genetically identical to each other when they're in a big colony like this. We see some corals that we call solitary corals, so that would be one of these polyps that lives all by itself. But these are clonal, so this is a colony of all these individual little polyps that are genetically identical to one another. That's really beautiful.
Between your cup corals and these octocorals is the octocoral tentacles are pinnulated, so you see those little side branches, and that helps close down the space between those tentacles and presumably allows it to capture smaller particles.
Yeah, really stunning. Absolutely stunning colony. That's really enormous. Yeah, and even, as we come back from it, too, I look at it again, and I think, ooh, primnoid. Of course, it's not. It's that bamboo coral,
Yeah.
Beautiful.
That's why the close-ups are valuable. Thank you.
Yeah, thank you so much, pilots.
Pull off.
Can we look at the base?
Let's just get a shot of the colony, and then I can come in on the base. This guy's waited 150 years for his debut on our live stream.
On its debut on Deep Sea TV. Just a beautiful old colony. As you might have heard the pilots mentioning, this is potentially 150 or more years old. A lot of these corals, well, most of these corals in the deep sea really grow very, very slowly compared to tropical corals. And that's because these corals, unlike the corals that live in shallow water, these don't have zooxanthellae. They don't have a photosynthetic symbiont that lives in their cells, photosynthesizing and producing excess energy. These corals just live entirely on what's falling through the water column, and so what's falling from above. And of course, any food that's produced in the upper ocean and travels down to the deep sea is being constantly picked at on its way down. So by the time you get here, 2,682 meters, there's really not a lot of food left. And so as a consequence of that, these corals grow just in a few millimeters per year, very, very slowly.
Sorry, I'm trying not to jump in, but as we passed over there 2 or 3 times, you got a really nice view looking down of something that we refer to as multiflabellate. And what that means is that there are basically, the colony has grown such that it has kind of like independent fans that are one next to another. And so if we cross over the top of that again and you look down, the viewer can see what's meant by that term.
Yeah, beautiful.
Oh, it's like layered, kind of. Rather than single layer.
Yeah, really stunning.
Really impressive holdfast there as well. That's the calcareous part that is sort of glued to the substrate. And as the coral colony grows larger there, you can see that beautiful multiflabellation, the different layers of fans that are going on there. Beautiful view. So yeah, that very large white holdfast at the base, there's also tissue covering that, and the tissue is depositing calcium carbonate onto calcite onto the rock to stabilize it. And as you might imagine, the larger the colony gets, the bigger the base has to be to support it.
If you want to check out this guy right in front of me, and then we'll climb up that one. Does that sound good?
Sounds great.
Okay.
Looks like that colony is an old veteran around here, and I see lots of sort of broken off twigs and skeletons and fossils. And so, perhaps at some time in the past it had many more related neighbors that were growing here. And for whatever reason, that one's in a good spot and has been able to persist through times maybe when the current isn't quite as strong and there isn't as much food.
You definitely wonder, you know, some of these branches that we're seeing sticking out from the rock face are really thick and chunky, just like that one was. And so you do wonder if it was the same species that was here once. Wow, look at that botryoidal texture on this face coming up here. Really different.
Yeah. Yeah, just incredible seeing the little bit of sediment outlining each one of those botryoids on the surface.
This might be like,
botryoids are thought to form because these are precipitating from seawater, and the spherical shape is sort of a natural kind of shape that oxide slowly precipitating might take. It's just sort of when there's nothing else to influence how it forms. They like to form in these spherical orientations, and so you get these botryoidal patterns on the surface of the crust.
Watch your starboard side.
And each one getting individually outlined with a little bit of sediment in the grooves in between.
See that?
And some of those abrasions, vertical abrasions in other parts of here, creating those kind of striped effect where sediment mixed with water has flowed down and eroded away some of the crust and left behind these vertical lines of sediment.
That's a wild feature.
Wow, just that view across from one of these little pinnacles to another. That's really amazing. You see the surface is dotted with these sponges that really pick up the white in the darkness when you shine light down here in the deep ocean and shine it out into the blackness. The whites really pick up, and so you can often almost instantly see these white corals and sponges before you can see any of the other animals that live down here.
Let's look at whatever's closest to you right now.
Okay.
Wow.
Yeah, I think it's the one you're looking at.
Yeah.
I'm coming to starboard with you.
And all interspersed with kind of fragments of skeletons that have fallen, these coral skeletons that have fallen down and died. You can see one kind of at the base of this long sponge that's just coming into view now.
It's a huge sponge.
Oh, there's another one of these big kind of trumpet-like sponges.
Yeah, so it's kind of like a ridge now.
Yeah.
And that's it. That's you, I think.
Yeah.
Safe distance. Quite the feature.
So this big white object kind of coming into view now is a sponge, and you can see those two red dots. Those are our harmless laser scales. They're 10 centimeters apart. So you can see how big this sponge is. Not quite as big as that coral we were just looking at, but still a very big object for down here in the deep ocean. We saw some of these a couple of dives ago. Christopher Kelly says that this is maybe a Conolasma species of sponge. They kind of have this beautiful funnel shape with all these ripples up on their face, up on its face. And looking behind the sponge here too, you can see this holdfast. It gets kind of narrow and attaches to the rock there, and covered with all kinds of fossil corals too. It's really landed and grown in a place with all these fossilized coral branches. Some older than others, where you can see it's brown. It means that ferromanganese is not precipitated out quite as much as on the ones that are this really dark black color. Beautiful, beautiful specimen. It's a really good look at the sponge. Yeah, it seems to definitely be growing out of all these fossilized coral stalks.
Let's get the fish.
Ah, fish. This one looks a little bit different, has a slightly different head than the two that we've seen, the ophidids that we've seen so far. Yeah, very different head shape. This one seems to have very large nostrils too. A lot of fish and other organisms in the deep ocean have very good senses of smell, because obviously there's not much light, not any light down here for them to be able to see, and so having really good senses of smell is a way that they're able to find each other, so they can find mates and find food. These are some great shots, pilots. Thank you. Christopher Kelly has put out, maybe this is a Bassogigas. But yeah, definitely a different fish. I don't believe that we've seen one of these so far in this expedition.
Just curious.
More kind of a humped back there.
Look how good that new tail camera looks.
Thank you, pilots. That was great.
Pretty well in the low light.
You might be getting close with your arm on the port side.
Port swing arm.
So certainly as we're coming up this this really wall feature, we're certainly seeing we're getting into an area of certainly a higher diversity of organisms. We're seeing more species of sponge, and we're certainly seeing some different species of corals here as well.
Once you
several different sponge morphologies here. So rounded, smaller species, and then these larger kind of flowing species here as well. Interspersed with some corallium I see in here. There was a couple of black corals earlier, some bamboo. Our depth right now is 2,663 meters.
Yeah, we might want to push more west.
Kind of following this real ridge feature up this wall. It's really been a fascinating wall to transit up.
Yeah, I'm curious. This spire is kind of sucking us north a little bit.
It's a really interesting chute feature here as well.
Once we get to the top
all the sediment in the center, and all of these fossilized and dead coral skeletons all accumulating in this chute.
I think we can do 10 meters. Let's see
what's that?
270.
Some truly impressive igneous flow features here. You can see
we're trying to go up
those nice
right?
flowing textures in some places, and then other places where it's been broken up by downslope debris.
Kind of like
really a lovely landscape in terms of rocks.
Kind of awkward.
Super awkward.
Yeah, it's really steep feature. We've seen several different types of habitat for different organisms. You know, we landed in that area that was sediment waves, and that's obviously very, very different from what we're looking at right now. And so we're seeing very, very different organisms.
Up the slope a bit, keep that. I mean, maybe what you're hitting right now, like 305.
Yeah.
Maybe do something like that.
Okay. 10 meters to get us kind of past the spire thing.
Yeah.
Very, very steep feature.
What was that rolling? Okay.
So 10 at 305.
Yeah. See that.
Bridge ROV nav.
Looks like your focus pretty close. Yeah.
I'd like to request to move 10 meters.
It's kind of nice getting this overview right now and looking at the different areas of the steep feature. So the pilots are, have to be very careful when driving the ROV. We're looking for overhangs. We don't want anything above our head. And so it's nice having these two camera systems, the Sirius, which you can see on live stream number 2, and then of course the D2 system, which is the main camera. And so pulling away a little bit from the wall actually gives a really nice overview of this of the terrain that we're looking at. This super steep feature with these little saddles and boulders and pillows and just lots and lots of texture here.
Yeah, I feel a little bit better now.
Yeah, that was kind of a protrusion kind of spire. Okay. Yeah, you're at 2650. That's like right here on the contours. It's crazy.
This is really just amazing to look at, this feature.
A lot of tether left.
Yeah, just
2,651 meters down, and we have these cliff faces, and all covered in corals and sponges.
We're just waiting on a ship move here. Watch lead.
Perfect. Thank you, pilot.
Pulled up to get over this little spire that I'm on top of.
Do we think it dips down on the other side, or do you think it's part of the face?
So below me, there's like a little fan that sticks out.
Okay.
And that's what we were looking at earlier.
Yeah, we climbed up that spire.
We climbed up this spire. So we pulled Sirius above it so that, because otherwise it was going to be like on Sirius's starboard side.
Yep.
So we're just waiting for the ship to move so I can get back and reacquire the wall.
Perfect. Thank you so much, pilot. There's a great little view looking down on where we just come from. We kind of worked our way up this spire feature and are now just working to get close to the wall again, having worked our way up that spire. But it really, you can really see the texture here, these sediment chutes that are running down and these larger boulders. This is really fascinating, Kira.
Yeah, and some of those, it's hard to tell when you're close, but you can really see this flow type morphology. As we zoomed out, it starts to make it more clear that we're looking at igneous features versus the wave-cut carbonate platforms we've dived on in the past 3 dives before the mapping break. And so we're definitely seeing the 2 major rock types that we find out here on these seamounts at these different depths, and here getting a better view of the volcanism in this region.
Yeah, the contrast between
Kevin
the wall.
Conrad, a volcanologist, in the chat. Sorry.
No, go ahead. Go ahead.
Yeah. Sorry for folks from home when we accidentally talk over each other. I'm actually doing my lead geologist annotating from shore, so I'm on a small delay, and so sometimes it's hard for Rian and I to hear each other, or if I accidentally speak over the pilots as well. Just letting everybody know that's one of the great things about the telepresence abilities is that Jason Chater and I were able to do this lead geology, help out with the crews from shore just to help with COVID protocols. And so I was just saying that Kevin Conrad from the chat was adding that these might be some low bait flow structures, creating these nice lobe-like textures, and maybe even some flows detached and sloped downhill as a whole. So you can have a whole flow sort of detached from the hill and then move together. It's hard to tell, so it's a bit speculative, but you can start seeing the stories that geologists tell when they see a view of a wall feature like this from this far out.
Yeah, it's views like this too that I think really drive home that these are really mountains under the sea. You know, these seamounts are underwater mountains, and you see these very high slope areas as well as the sedimented areas like we landed in, but they're just like mountains up on land. You know, they have different morphologies at the base than they do at their top.
Get this guy real
It's
quick.
really, really fascinating. This is a sea urchin. This looks like a Okeanos, a white sea urchin here. I think it's sitting on an old coral skeleton there with zoanthids polyps on it.
Rian, I wanted to continue your analogy of the mountains, and you know, just like on land, as you travel from the base of the mountain to the top of the mountain, you see the community around you change. The most obvious of that is if you're on a really tall mountain, you get above the tree line, but as you approach that tree line, the trees tend to get, you know, shorter, squatter, dealing more with the wind and so on. And so we see the same thing as we scale these seamounts. Now, because of the rate that we're able to go up during any one dive, we don't tend to see that really dramatic change during one dive. But as we accumulate the different dives, we see these very different communities in the different depth ranges.
Yeah, absolutely, Scott. Absolutely.
Yeah, and Kevin Conrad adding from the chat that these low bait structures form from the lava quickly cooling upon its eruption. So when we have these underwater volcanoes actually erupting at the seafloor, they can form this crust, and then more lava gets pumped into the flow and goes beneath the crust and causes the lava to sort of balloon up from the inside. So you can start to imagine how the igneous processes were forming these really interesting, large, smooth, loamy textures that we're seeing here. Thanks so much for that, Kevin.
Yeah, and here we see on this kind of smoother area some more of those, they almost look like scrape marks coming down, as Jason Chater was telling us the other day, that these are areas where water becomes supersaturated with sediment, and they fall down kind of the sides of these cliffs and cause these kind of scar marks that will eventually potentially become some of these chasms that we see as they're eroded away through time. This is an interesting little collection of corals on these dead skeletons here. Chrysogorgia and a corallium, and lots of this coral skeleton, this dead coral skeleton, with actually sediments kind of caught on top of those dead coral skeletons too. It's interesting too, Kira, that, you know, looking at this image right now, kind of in the center is very smooth, and then you have that botryoidal just off to the right-hand side. You know, do things like water currents affect where these ferromanganese crusts actually occur? Like, would the water have been flowing faster and stopping it actually precipitating, or maybe the other way around?
Yeah, that's an excellent question, because these are chemical sedimentary rocks, or coatings, I guess. Ferromanganese crusts are definitely influenced by water masses, both their physical properties and their chemical properties. So in a region like that, absolutely, as sediment-rich waters and fast currents go past, it can start to erode away the surface of the ferromanganese crust, and then on another face of the rock where the currents still keep it free of sediment, but it's not as intense as to erode it, you can get a really thick crust and really freshly forming crust precipitating there. So the morphology of the crust certainly tell you something also about the water masses, how they're moving around, and those kinds of things. And importantly, when we see those crusts being eroded, we're removing millions of years worth of the seawater chemistry. I feel like I'm tongue-tied today.
Yeah, this is like a gelatinous house of something that's been living in the upper water column that's falling down. So there are organisms that live up in the upper ocean that create kind of a mucus housing to help them capture food, and eventually they become clogged, and the organisms will shed those houses, and those houses actually falling down in the deep sea are actually a really fundamental part of what helps to feed the deep ocean. They're great sources of carbon for the organisms that live down there. So I think that's what we were just seeing just then, a kind of a mucus house of some kind of upper water column organism falling down. See this little chasm here filled with sediment, the chute.
We can probably get 10 more meters in.
Yeah.
Bridge ROV nav.
300.
Hold one, please.
Oh, great shot here looking up.
Did you want to change it to 300?
Yeah, let's do 300.
Okay.
That's my current heading, Josh. 300.
Yeah, it's almost like a little vertical knife ridge. Or, I mean, this is an interesting outcrop of rock and lots of things living on it.
10 meters, bearing 300 degrees, speed decimal 2 knots. Good copy, thank you.
And these edges like this can actually enhance the water flow a little bit, so kind of funnel it and squeeze it in between these two faces, and so that's often why we see a lot of organisms living kind of on the side of these features. Piles of dead skeletons there, all kind of accumulating on this ledge.
Let's come in on the purple guy here.
Oh, well spotted. This is a little sea cucumber. Yeah, different from the one that we were looking at earlier. This one's very smooth, so it doesn't have those little pseudopodia, those big tentacles sticking out off of the top. But this is another sea cucumber. And you can kind of see inside the gut system. A lot of these sea cucumbers are actually fairly transparent, and so it's kind of fun that you can see what's happening on the inside. And so these sea cucumbers eat sediment, and they have to eat a lot of sediment to get the nutrients that they need, because there's not a huge amount of nutrients in sediment. And so they have very, very long and twisty gut systems where they ingest the sediment. The sediment moves through slowly, and so has lots of time to get all the good stuff out before it's pooped out the other end.
Coming out.
Thank you, pilots. All these big, big, long branches here. And coming across the other side of this chasm, here we have, you can see some Bathypathes, and I've been corrected. The corallium is actually now moved into the hemicorallium. Even though the genus corallium is still valid and has at least 7 species, but the species that we're seeing today have been moved into the hemicorallium. That happens fairly frequently in the deep ocean. We're discovering new species, or species that are actually the same and should belong to the same species or the same group. And so very often species in the deep ocean change names as we discover more and more in the deep ocean.
Copy, thank you.
Let's do a quick zoom here.
You can see this large stalk sponge with these lobes, and actually this one looks like it's been covered over, or is it died? I think that's the skeleton. So where these sponges die off, so they can leave behind a skeleton. And the skeleton is often this brown color. So I think this is a sponge that's partially dyed. The white is still alive, and the brown is where it's died off. But these sponge skeletons can actually form important habitat as well. You can actually see all these, there's some hydroids on there and some forams living on the skeleton of this sponge. Beautiful. It just provides structure. So organisms that provide structure are often called ecosystem engineers, and so those organisms are creating ecosystems for other organisms to come and live. And sponges and corals often fall into that category of being ecosystem engineers. You can see this is mostly dead coral, and then this little pop of bright pink of this hemicorallium in the middle here. Oh, there's some white over to the left as well, just living inside all these skeletons. And it'd be hard to tell from this mess whether it's some of the original coral that still survived and is living, or whether it's a different coral that's come and landed. The larvae has landed and started to grow in this area. There's some white there as well. Actually, I think the white is one of the original colonies that's fallen over. You can kind of see the dead branch, and it's branching out white at the very tips. Just the last little bits of that one alive.
I think also possibly, Rian, there may be, I was going to say there's maybe a stoloniferous octocoral, which would be like a ribbon or mat-like one, but I may have been fooled. It may have just been
there may be some up to the top here. Yeah, there may be some of that up on the top here. There's definitely a few different things going on. And some sponges in there as well.
Definitely a lot of life taking advantage of this old skeleton, this this habitat that has been created by this organism.
Yeah, absolutely, and why not?
Yep.
The yellow loop.
This is beautiful. Another really large colony here.
Interesting. It looks like two, I'm going to say Coralliidae, the family Hemicorallium is in, and I'm not certain if the white one is different from the pink one. I can't say with certainty at this point.
Yeah, beautiful images of the polyps there.
One of the things that's interesting in general about the Hemicorallium and that whole family and the group that it belongs to is the structure of the skeleton is very different from the other octocorals that we see here, the bamboo corals, the chrysogorgiids, the primnoids, and so on, in that these are more, I think of it more like concrete or cement matrix, where the sclerites and some tissue, they're all sort of glued together to form the branches that you see, instead of having a solid single internal axial skeleton that the tissue grows around. Instead of in this type, the tissue is embedded in with the skeleton. It's a very, very different sort of construction.
Yeah, very different kind of octocoral.
Got a little bit of tether, but I can
this is a beautiful sponge. That one looks a little bit different from anything that we've seen so far.
Okay, get a little
Sorry, and if we're still here, Rian, if you look just at the lower part of the screen, maybe a feeding trace there? Is that what I'm seeing, all that black?
Oh, that might be, yeah.
On the rock?
Yes, that might be.
It's been an urchin. Actually, I may see an urchin. I think I see a white ball at one point there in the upper left.
We saw some of these with chitons as well earlier in the expedition.
Oh, yeah, chitons. Right.
So I actually wonder, I think we're looking at the culprit right now, but I don't think that's a chiton.
I'm going to come in on it a little bit.
I just got to still wait for the zoom.
Let me track with you, go and pull out.
Yeah, you got a little tether now, so.
That looks more to me like there's something sessile that's growing on the exposed rock and not.
You think so? Okay.
These,
I don't know, it's right at the end of the traces.
Watch your left toe again, yeah.
Yeah, I
Below
agree.
your left toe.
It's got a little split shell.
Almost looks like a bivalve, doesn't it, from this perspective?
It's got a gelatinous material around the outside edge.
Yeah, that's right, Roland. So it has kind of a gelatinous material on the outside.
A slit snail, I think. Slit shell snail?
Yeah, yeah.
Wow.
Really unusual.
A great observation, yeah. I mean, that's got to be what's
it.
making that feeding trace. It's,
Trying to get
you know, well, these snails have radula, which is a hard protein rasping tongue. It's full of spines, and it, excuse me, it's on a very muscular structure called the odontophore. Think of your tongue. If you're able to extend your tongue out of the mouth and press it against the surface and then scrape it back and forth like sandpaper, and it scrape up whatever bacteria or other stuff is growing there.
Oh, that's what's
and that's what it pulls in.
I get it.
Creates these mucus strings that bind up those particles and get it into the mouth and live on that.
Yeah, this is a good
Outstanding observation.
This is a good shot here. Yeah, this is a really unusual to be able to see one of these. A slit snail shell. Slit shell snail.
Sorry, might be some little tugs with the heave here.
It makes me wonder, too, with the ferromanganese coating on the rock, does it end up eating some of the ferromanganese? And can that be a nutrient for them or a good electron exchange? You know, a ferromanganese crust create a really nice electron environment for lots of microbiology. So very interesting to consider what kinds of benefits they might get or detriments they might get from also accidentally ingesting some of the crust material. I've never seen this before.
That's really interesting, Kira, and I wonder, I could see a role for some of those minerals to be played in radula itself, which is that tongue-like structure. So it would be fascinating to look at those
very small
details.
Yeah, so the pilots are going to take a look and see whether it might actually be possible to sample this one. This is a really unusual observation in the deep sea, and it would be really fascinating to learn what species this is, or even describe a new species if it is.
We can ask science which one makes more sense. What size filter should we put on here?
I think we can go, how big is it? It's a couple of centimeters. Yeah, so I think the coarse filter is fine.
Coarse, okay.
Yep. I think you're going to need the extra suction.
Okay. Say bye, Ron. Get your suction light all the way up. There it goes into color mode.
It's so interesting to me, the number of times that we're down here, the observations that we've made, and seeing these feeding traces that we've never seen in one of these split shells doing this. So every observation is such a valuable one.
Oh.
Indeed, Scott. Indeed.
All right. You mean wing inboard and then draw it further?
Just bump it a little bit out. I might be able to grab it.
Okay. Try to give you a small, just ever so slightly. Okay, off stick. Can you draw back in?
Yeah.
Okay. All right, jar 2's ready to go. let me know when you want the suction motor on and what RPM. Start at 1,000 maybe. A little less, 800.
Oh, there it is. Yep, you can start it up.
Okay. 800, 1,000. Positive RPM. Okay, reducing RPM just a little bit. 700 RPM. All right, indexing.
Really nicely done, pilot. Thank you so much.
All right, jar 2. Okay, I'm going to draw back out a little bit.
Yeah, a little bit.
Okay, draw coming out. Easy.
Yeah, that was a really exciting collection of something that's really unusual here in the deep sea. A great discovery.
Nice. Okay, retracting drawer.
You know, another thing that's so valuable about that collection is that, you know, if, let's say, slit shells are fairly common in the deep sea, you can see that they're adapted here for scraping the hard substrate. You saw how small that was. That is just something we can't collect with a dredge or a net. The only way to be able to get it is using the technology like we just used. And having that connected to this in situ video has already told us something about its biology. You can see the time that it is spent right here, meandering around the rock and scraping the rock. And then those really interesting questions that Kira asked about, you know, well, is it using some elements in the crust and so on? So there's an awful lot we might be able to learn from that single collection.
Absolutely, Scott. Absolutely. Yeah, just from this one collection, we know a little bit about its ecology and how it's feeding and where it was located on these walls and at depth. And as Scott said, if this had been collected in a dredge or a trawl, the chances are it would have been crushed and not be able to be identified by the time we got it to the surface. So this is just an amazing collection and observation here on this wall.
Okay, I'll center back up on you.
Okay. Yeah, the closest thing to me is what you're looking at. Okay. So we can do 10 meters, 290. Does that sound good? And then we'll likely get to the top of it and turn to port towards more the west. Does that sound good?
So 10 meters is going to put that pretty close to you.
It's at 25 out?
Yeah.
Okay, we can come up first.
Yeah, we'll come up first. And then
I think it's going to be like a local high, and then we're going to have to like reacquire where the hill is.
Nice work on the suction.
Yeah, so that's what you see, and then
I'll come up and we can see what's ahead of us. There's a sponge that looked interesting over here, I think, too.
Okay.
Yeah. So, oh yeah, you can see it if you look at the sonar.
Oh yeah, there it is.
I'll just do a quick scan.
Paint it.
Yeah, so there's another.
It's kind of like vertical.
Like another like peak there with a shadow, and then you can see behind it.
Okay.
And that's 20 meters away. Yeah, so you could, yeah, see how it dips down and comes back.
Yeah, this is like a local high, and then it dips down, and then the wall.
It's like another little pinnacle here that we've been working our way up. There seem to have been a few of these on our way up this this wall feature. If you're just joining us, I'd like to say welcome to Dive 12 of the NOAA Ocean Exploration Expedition North Atlantic Stepping Stones. I'm Rian Waller, the lead biologist for this expedition, and Kira Maisel is on the telephone line as the lead geologist. And today's stepping stone is in the the eastern end of the New England Seamounts on an area that was previously unmapped and is unexplored. We called it Y Seamount, nicknamed it Y Seamount. There's a little Y feature on the top. And today we're diving on kind of the western peak. This is a two-peak seamount, and we're diving on the western peak up a fairly steep cliff face here. And I've really been through some really interesting geological morphology and seen some really fascinating biological changes as we've been making our way up this wall. This is a really fascinating sponge here. Again, a little bit different from things that we've seen so far.
Whoa.
Hey, come on in.
I've never seen this. It's an interesting looking sponge.
This sponge almost looks like it's been gone over with an iron. It's so smooth. You can see the little pores all over these sponges. So these sponges are filter feeders. So they have all these pores on their outer surfaces that let water in. They have specialized cells called choanocytes with little flagella that wiggle, and this wiggling creates water flow.
My, upper swing arms down.
Sure.
A little bit.
Yeah, unusual sponge there. Very, very flattened.
Base.
Do you get it?
Yeah, that's good.
Okay.
That's great, Pilot. Thank you.
Go right around it as you leave. Show the edge on.
Just masses of these old coral skeletons, some attached and some fallen down.
So thin.
It's very thin. I mean, it really looks like it's been ironed.
Am I tugging you?
Yeah. Must be.
Yeah. Copy that. Sounds good.
Yeah, you can fix that upper swing arm if you haven't already.
Upper swing arm, starboard, coming back up. And it's up. Hydraulics disabled. You want 10 meters after that heading change?
Yeah, after the heading change.
We do my heading 285.
Yes, that's great.
Okay.
As we come back from this wall just a little bit, you can really see the topography. And if you change over to live stream number 2, you get a great view from this Sirius camera. This is a camera on the second part of this two-body ROV system that's looking down on us. And you can see how steep this face is. And we've been going up various different pinnacles as we're making our way along this dive track. We're currently not too far off the top of a wall feature and hopefully coming up onto a plateau to look at a slightly different part of this feature. Be great to get a zoom on that coral rubble if it's possible, Pilot.
On the lower?
Actually,
on the
almost.
ground or on the wall?
Actually, on the wall. Yeah, attached to the wall, kind of central. Thank you. Yeah, so these corals, I can see a species called Desmophyllum dianthus. These are solitary cup corals, and there's actually a lot of them here. I can count nearly a dozen. Yeah, there's definitely more than a dozen here. I'm not sure I see any other species. This just seems to be layered cup corals, one on top of the other. And where we see these cup corals in these really thick deposits like this, these weren't all alive at one time. There was originally one layer, and as those corals died, other larvae came in and settled and grew on top of those, and then same thing happens and you create another layer, and so you end up with these really large accretions of these cup corals all growing off of one another. And some of these cup corals from this region have been dated, most of them date at around the last glacial maxima, so about 20,000 years ago. But some of them have been dated back 100, 200,000 years. Was that little egg case in there? Or is that a tunicate? Hard to know, it's a little far away. Not quite sure what that one is. I think it's a tunicate or sea squirt in a colony. Kind of encrusting on this old coral skeleton here. Yeah, this is a tunicate, a colonal tunicate. So similar to the corals, tunicates can come as singular organisms that are genetically distinct, or they can come as colonies where they're all genetically identical. Thank you, Pilot. That's great. Oh, there's Desmophyllum dianthus there, all fossilized over.
Ian, I thought perhaps that tunicate looked like a collection of individuals together rather than a colony. I'm not certain. I totally agree with what you said.
So I could kind of see a little bit of a mantle on the base of the rock. That's why I think maybe it was a colony. I could kind of see it starting to encrust at the base. But I agree, each of them looked very distinct initially there.
285.
285.
It was at least not a compound tunicate.
Bridge ROV nav.
Each of the units was clearly
Like
had its
request
own,
to
I think,
move, range 10
Yes.
meters, bearing
Nice little
285 degrees, speed
Yep.
decimal 2 knots. Good copy, thank you.
Over to port, see if I see anything. Just waiting on a ship move, watch lead.
Oh, great. Yeah, in that case, we should definitely do a few zooms if we can.
Got your blue view.
Definitely very blocky terrain to go over.
Yep. Anything in particular?
Yeah, let's have a look at some of these sponges. So, where are the lasers? Yeah, this large one in the center is good.
Just come on in.
A lot of structure in this sponge, lots of little lobes and hidey holes for other organisms to potentially live. There's two different kinds of sponges in this one shot here. So there's this more encrusting type sponge on the bottom, and then this whiter, brighter white, more structured sponge up above. And these sponges, like corals, form structure on the seafloor for other organisms to use as their habitat. And we get a lot of organisms, brittle stars and worms and snails and crabs, all kinds of other smaller organisms like to use sponges and corals to be able to live. They provide protection from predators, to get out of the way of predators and hide. They also perform protection for laying eggs or harboring young. And they also provide great places to feed as well on all the other organisms that come and live around these corals and sponges. And although, you know, this ROV dive is really kind of a snapshot in time, we get an idea of what's going on right at the moment. But these corals and sponges can also form habitat for more mobile fauna as well. So things like fish might come and use corals and sponges for laying eggs or providing areas to feed as well. And we're just not seeing them right now because they're mobile and they move. So it's really important to remember that these ROV dives are really that snapshot of what's going on right at this moment, but something else could be happening in an hour or two. This is potentially a rosellid sponge that we're seeing, kind of this spiny vase. Thank you, Chris Kelly. And yeah, this little old coral reef structure here, this paleo reef of this underlying corals, and then some newer species that have grown up on top along with these sponges. And then a relatively flat upper surface with just a few colonies poking out, as opposed to kind of this pavement of this paleo reef.
Let's get this thing in the water.
Oh no, that's cute. This is a ctenophore. You can see kind of the glowing teens, and that's what I was looking out for. You can see them kind of flapping their rows of their comb rows, or called teens.
I'm going to come in on it a little bit.
And these ctenophores are actually predators, so they can actually swim through the water. Unlike jellies that are mostly pushed by water currents, ctenophores actually have the ability to swim and move against the current a little bit. And we heard earlier in this expedition the red bellies of these ctenophores are ways to hide prey. So the ctenophore is transparent, and so when they eat organisms, you'd be able to see what the organism they'd eaten inside, and that could make them a potential prey item themselves. But by having a red belly, they actually enclose the prey that they've eaten and in this red casing. And red is invisible in the deep ocean, so red light is attenuated very, very early on as you go down in the ocean. And so by the time you get down to these depths, if you're a red color, you're nearly invisible. It's really beautiful. A very unusual shape too. Not a shape that I've seen, and looking in the chat room here too, you agree this is an unusual shape for a ctenophore. It's really quite beautiful. And the teens that you're seeing, the comb rows there shining and glimmering in the lights of the ROV system, are usually kind of on the head end of these ctenophores.
It's very close.
Beautiful. Really good detail there. Seems to have these large lobes.
Yeah, I was equating those lobes to lips. The mouth would be in the orientation we're looking at now to the right. So just to the right of that pigmented stomach that you were
just float off my starboard.
And those lobes can help force the prey in towards the mouth.
Coming back at you pretty quick.
Yeah, just let them go.
Really beautiful. Thank you, pilots.
Full line.
Yeah, pushing ahead. Coming up. Pushing ahead.
Okay.
See if we can make the jump. Yeah, there's a lot of stuff on the backside too.
There is a lot of stuff on the backside. Look at the backside of this pinnacle and this little chute that's going down it. You see some chrysogorgia, some more of this hemicorallium, potentially geodia sponges, these round bowl sponges that are slightly brown. Wow, and it's just amazing looking down there.
You want to get another 10 meters so I can kind of get 285?
Yeah.
Bridge ROV nav. I'd like to request to move 10 meters, range, bearing 285 degrees, speed
Get more sediment here on areas that are not quite as steep. And then a lot of these coral skeletons that have fallen down from above and kind of accumulated in these areas.
Good copy, thank you.
Holes. It's definitely been very interesting today, checking out a little bit more of the igneous geology of the region. Seeing these really big flows in this vertical cliff feature. We chose a dive track today with sort of two little peaks or two little plateaus going up. And you can see on the map in HYPACK that we're making our way up the steep feature, and then we're going to get to a little ledge, and we'll be able to see how much sediment has accumulated and who's hanging out on the ledge. As you can, that is sort of halfway up this ridge feature, all the way up to the very top of what we're calling Y Seamount. Definitely here seeing igneous rock covered in ferromanganese crust and some remnants of old corals. We're at the easternmost New England seamount that we'll visit on our way back to shore in a little region we're calling Y Seamount. If you check out feed 3, you can see how far we are away from the coast. Lots of seamounts to visit as we continue our journey, but enjoying seeing the connectivity between the further east cornerized seamounts. And I'm sorry, I feel like I maybe kept saying east, and I meant to say we're moving west towards the east coast of the U.S. So pardon me if I was saying the wrong thing. I know what I mean, but I'm saying the wrong thing. So we're slowly moving west back towards the east coast of the U.S. and enjoying this easternmost seamount in the New England seamounts as we make that journey back. Zooming in here.
Hi, this looks interesting. I think it's a sponge,
yeah, this
but
looks like...
Oh, go for it.
Yeah, Kim, for sure. Yeah, absolutely a sponge, perhaps in the family Fariidae. One of the glass sponges. Yes, Chris Kelly and Tina Molotova would agree with that assessment. That is a Fariid. They grow with these really ruffly patterns, which is one of the ways that we can make this ID to the level of family. But then they can take on a great number of different shapes with those ruffles. And so you see this one is kind of tall and elongate like a single stalk, but they have all kinds of morphologies around that. And I should say to listeners who are following along and enjoying some of the organisms here and thinking about some of the names, we do have a benthic animal guide available on the Ocean Explorer website. If you would like to sort of go through that, not necessarily to study the names. Oh, by the way, there was another snail, another gastropod snail down there. This one was different. It's a predatory type snail. Have a look at the benthic animal guide just for some of the diversity of shapes of all of these different sponges and corals and so on. Just a wonder to behold, and a great product that was put together by Chris Kelly and his team out at the University of Hawaii.
Yeah, thanks, Scott. There was a lot of effort put into that benthic animal guide, so we're hoping to continuously expand it. And it looks here like we have another one of those large sponges that we had seen out in the cornerized seamounts. Trying to remember the name, but if someone on the chat room could chime in or on the call.
Yeah, I think it was Conolasma, and we did see one of these earlier today as well. They have these beautiful large vase shapes, and perhaps as we turn around to the side here, you'll see what a great sponge, a beautiful sponge this is. They can get these enormous sizes. It's another interesting theme, to explore along with all the corals that we're seeing is all of these sponges are essentially feeding in the same way. They have internal cells that can generate a current. They have small flagella in them, and those flagella are constantly beating. And that means that the water that is near the surface of the sponge is pulled in by suction because of that beating. And any small food particles that are in there can then get captured by structures on the inside of the sponge at the cellular level, and then the water is sort of moved away from there. But what you can see is that there's just an incredible diversity of morphology of the overall sponge in which that functional construction is embedded. And it's really hard to answer the question about, well, why does a sponge have this shape and not that shape? Earlier today, we were seeing these ones that sure fooled us into thinking they were corals when we first saw them. Looked like these nice branching antler-like structures. And there you go, those nice ruffles in the background, the large vases, and so on. But they're all basically feeding the same. And so there's lots of different ways to deploy that functional biology in a larger body.
As we said earlier, Scott, another mystery of the deep sea.
It's going to keep all the deep sea biologists in business for a long time.
That's good. Hopefully you can get more grad students then, too. There's quite a bit of coral rubble now in this area. Oh, look at this sponge. These variety of sponges, and Scott was just mentioning how we really don't know why sponges grow in certain ways that they do, and we're not even sure about how growth rates of sponges. I'm not sure if we know about any kinds of sponges of growth rates. It's still quite a mystery. And there are these small ones that almost look like polyps of sponges right next to this really large bumpy sponge is what I can describe it as.
Go to one of the small ones.
Yeah, I described it as a cauliflower-like sponge. It's like bunches of cauliflower all grown together, and I suspect that that's a different species of sponge, those smaller ones that you're referring to on the left side. I tell you, as I see, the range of crazy shapes and sizes and things in this landscape that we're seeing, I sometimes think Dr. Seuss had an ROV way before the rest of us and got some of his ideas for his crazy environments from what he saw down here.
I agree. That must have been something that they didn't tell us. There's a lot in the deep sea that looks like it came straight out of Dr. Seuss. And there's a lot of these what look like Geoidea sponges. I don't know if you can correct me, but these kind of more bulbous, darker colored sponges that are kind of in between some branches and clasping on in various of these dead stalks.
Bridge ROV nav.
Yeah, Kim, I would agree. That's
like to request to move range 5 meters.
I can tell you that in our chat room, Chris Kelly is probably has the most expertise in identifying sponges. And he was completely stumped by that cauliflower-like one and those little bulbous ones that were to the left of it. So, the biology continues to surprise and amaze us, and there's so much undescribed diversity that's down here.
Absolutely. We have quite the geomorphology in this area. Kira, looks like it kind of changed. We're having less and less sediment and more of these large lava kind of pikes.
Turn to starboard.
Yeah, absolutely. As we've gotten more vertical and we've gotten into the steeper part of this and no longer down at the base of the cliff or the slope, we're seeing these less sedimented, more exposed pinnacles or pillars, like you're saying. It's definitely still coated in ferromanganese crust and still appears to be igneous rocks, but the change in where it is in this slope feature has really given us a different view and a great habitat as well.
Pilot, could we get a snap zoom on that center stock kind of poking out right at the center of the screen? What looks like a brush, a bottle brush?
Come on in.
Yes. It looks yellowish. That's all I got.
It's not the coral.
Looks like a coral.
Yeah, this is interesting. Yeah, as we were zooming in, I was just going to, quote-unquote, dismiss it as a zoanthid, that is a colonial anemone overgrowing skeleton. But in fact, this is an octocoral. And so I am going to suggest that what we're looking at here is an octocoral that is growing kind of like a mat or a ribbon, overgrowing another dead skeleton. But boy, it sure is thick in there.
This one's really fascinating too, because that skeleton looks really old. Like doesn't look like a fresh.
That's why I think it's overgrowing.
Yeah.
It's either a stolonifer or in the family Anthothelidae, which does do that sort of thing. Yeah, there you can. It's growing as a ribbon along the side of that skeleton. So this coral, these polyps did not create the skeleton, as you know. That's a really old skeleton. So it's overgrowing. And it's really hard to see for me. Maybe you can see it better. I can barely see the connection between polyps.
I actually can't see the connection between polyps. This is why I'm sitting here trying to stare at it and see if they're connected.
Gotta be there.
It just must be a very, very thin and fine coating. But yeah, even on these big screens, I cannot see it.
I mean, as far as I know, in the world of the 3,000 or so described octocoral species, there is only one that grows solitary, and that's known from off of deep New Zealand. It's a really bizarre thing about the size of a thumb that lives in the soft sediment. So this has to be growing as a stolon or a mat, a ribbon. That is, there's a common mass of tissue that they're all arising from. And you can see that easily towards the end.
Yeah, this top end, you can really see that. Yep. Thank you so much, pilots.
I'm going to say that this is in the family Anthothelidae, because the other stolonifers shouldn't be able to sort of recreate that solid coenenkine mass, I don't think, which is the name of the tissue between all the polyps. Very interesting.
Yeah, really interesting.
Yeah. So you're just finishing 5 meters, correct?
That's correct. There's the larger wall back there. I haven't been looking at. There it goes. Yeah. Sonar's got it. It's pretty straight. Perpendicular to it. It's still a little bit northbound.
So it's 10 meters off from me.
Okay.
So you could probably move.
It's interesting taking a peek at feed 2. You can see camera 2. You can see this sort of vertical igneous dike or sort of pinnacle that we're diving on in the context of the region. It's sort of definitely a feature that's sticking up really high compared to the rest of the igneous flow around it.
Look on the south side of this.
Okay. You want to wait on the move then?
No, you can call it in. It's going to take a little bit.
Okay. A 280. degrees would be good.
Bridge ROV nav. I'd like to request to move range 10 meters, bearing 280 degrees, speed decimal 2 knots. Good copy, thank you.
Kevin Conrad in the chat, a volcanologist, was just giving us a clue that what this feature could be, that, or the one we were just recently on that was sort of sticking up, could be a parasitic cone, which is a small volcanic cone that forms off the main magma body. There's some great diagrams of it if you Google it. I have never heard of that before, but it makes sense. It'll pull a little magma off the main chamber and create a separate little dome.
Not sure.
Hard to tell if that's exactly what it was, but would explain why it's a tall feature, sort of jutting out away from everything else.
Yeah. I'm sorry, I couldn't hear you. 1859.
Do a quick zoom here. Okay, let's come out.
On the right side of that hemicorallium, I noticed a kind of a muddy, empty bag-like structure, and I wonder if that may have been a vacated egg case.
On the hemicorallium, to the right side of the hemicorallium? So the bright pink in the center here?
The center of the screen.
Okay.
And it was on the lower right.
Oh, yeah, yeah. It's like a little...
round sack almost.
Thank you, Bridge.
Like a little mucus sac there. Yeah, and so what Scott is suggesting that this is potentially kind of the mucus outer of an egg case. We've observed octopi egg cases in corals occasionally, and other organisms lay their eggs in corals as well. And so this might be the remnants of one of those egg cases. It doesn't look like anything is home. It definitely looks like it's just the empty sac left behind.
Yeah. With that wonderful close-up, thank you. The alternative explanation is that was some kind of gelatinous organism that was moving through the water column and is now kind of dead and just accumulating sediment. That whip, I think, is new for the day.
But,
past it on the right.
Oh, sorry, I didn't quite catch it, Scott.
Yeah, no problem. We'll keep an eye out for it.
Oh, the straight up and down right there.
No, it went.
Yep.
Yep, it's so straight that I'll bet it's not a bamboo coral. I'll bet it's a primnoidae.
I don't know. My primnoid earlier turned out to be a bamboo. Which one is this?
Yeah, you can see the polyps are coming off in what we call whorls,
Mm-hmm.
clumps. Looks like there are 3 or 4, perhaps more than that, all coming off from a common area. And even more distinctive than that, from my octocoral taxonomy eye, is the large plate-like sclerites that are going up the body of the polyp. So unquestionably Primnoidae, probably the genus Calyptra. Very nice. That may be the first primnode of the day.
Yeah, I believe it is. It's certainly the first I have in my notes of the day today. And a little bit different from the Calyptra that we were seeing just a couple of dives ago, or even actually on the last dive, which had kind of that bend at the end. These ones are very, very straight up and down. Oh, it does have a little branch.
Sort of like shepherd staffs, weren't they?
Yeah, this one does have a branch at the bottom that's underdeveloped.
Ah.
Oh,
Well, that's interesting.
It is interesting. Thank you so much, pilots.
ID.
So I still have the spire kind of below me here.
Mm-hmm.
And
I'll
then,
set her up on you.
Yeah. No issues.
What's that?
I think I'm plenty clear of it. I'm way above it at this point.
Yeah, I came up, so.
Yep. I came up with you. I have some more room to come up now.
I still find it so interesting in this region of the global oceans, looking at Corner Rise and New England Seamounts, all of the coral rubble and its coating with ferromanganese.
Is that how thick the crust is?
It's so interesting, just that little bit of history it's giving us about when this seamount was shallower and the communities that used to be here. I'm not sure I've seen that as often in the Pacific or other places. It's definitely giving us some really interesting clues about the history of the seamount, and now kind of climbing up to a little mini plateau where we definitely have some down slope sediments and other debris, coral rubble flowing down over these really beautiful flow bait lava flows. And Kevin Conrad notes that for some of them you can see them cross-cutting each other, which means that you can see where the different lava paths have crossed over each other, and in some places you can even see where the lava crust has flaked off.
Cool shot. Serious.
Yeah, the pilots just noted it's worth having a look at live stream number 2 for the Sirius cam, looking down onto D2, and you get a real... Oh. I think it is. Interesting, because it does say that I am open.
Yeah, I can hear you.
Okay. So if it's worth looking at live stream number 2 for the Sirius cam, looking down onto D2, and you can kind of see the topography that we're in, kind of this very, very steep, steep area here. But we should be coming across this plateau, kind of coming to the top of this little steep feature, and then we have a plateau, and then another steep feature. It's kind of been noted as we've been working our way up that there's really quite a lot of dead colonies of coral as we've been making our way up this wall, both the older that are really covered over in the ferromanganese, but also relatively recent ones as well. And it would be really fascinating to be able to go back through time, to be able to look at coral colonies and see what this seamount looked like, you know, 1,000 years ago, 100 years ago, even 10,000 years ago. I think we'd see a very, very different set of organisms, or different species of organisms here than we're seeing today.
You want 15 meters? Or just 10?
Just do 10 for right now, and then...
Okay. Sounds great.
Bridge ROV nav.
Oh, here we definitely seem to have come to kind of the top of this plateau.
I'd like to request to move range 10 meters,
the topography has got a lot less steep here.
280 degrees, speed decimal 2 knots.
Kind of made our way to this top of this wall feature. And like so often when we get to kind of the tops of walls, we often see it really encrusted with organisms as these tops can cause kind of extra flow.
Do a quick zoom here.
Watch lead pilot?
Go ahead.
Yeah, I think, are you going to zoom in on the red?
Yeah. Is that jelly?
It
Oh,
does
yeah, it's one of
look
those
like
that's, like,
it's
stuck to the
jelly. I'm trying to see whether it's a siphonophore, but I think it's a jelly.
The one
Yeah,
that's
see-
got
The strands.
the anchors.
The anchors, yeah. Oh, it just released.
Isn't that one of the
Yeah,
dandelions?
it's kind of attached to the bottom there. But I don't see all the different different polyps.
Gonna come in on it, Roland.
I'll try to track.
Some suggestions from the
Sorry.
chat room, maybe a hydrozoan. Go ahead, Scott.
Yeah.
Yeah, I wasn't paying attention. I think this is
Should have some more
one of
tether
the
pretty soon.
dandelion animals, the, what is it, Ropalium? And it's just mostly released itself from the substrate, so that would make it a siphonophore.
Yeah, though I was trying to look at the different, the colonal, so these siphonophores are colonies, but I just, it's got this outer
Well,
covering, and I don't
colorful
see
lights and
those
getting
different,
closer.
I don't have any tether.
Okay.
different
Copy.
pieces.
Yeah, I agree. It's normally that colony should have a gas-inflated float in the upper center.
Yeah.
And you would be able to see the sort of swimming zooids and the feeding zooids and so on.
Mhm.
And they anchor themselves to the substrate using these tentacles. So they're very different from most siphonophores that are drifting through the water column. And, it may be something else, something related, and it's just kind of hung up on the bottom, but that is sure what it reminds me of.
Yeah, me too. It's just, like I say, it's just missing those really obvious zooids. Usually you can kind of tell. So these deep-sea dandelions are made out of, they're a colony of different zooids all together, each with a different job. And so some of them are swimming, some of them are for reproduction, some of them are for feeding. And so usually you kind of see those different structures all kind of doing their own jobs. But this one, I don't see that. It's really fascinating.
Yeah, there's some type of feature. Right
They say
there.
these long tentacles that usually keep them attached to the bottom. Thank you so much, pilot.
Something there.
Really interesting.
I think that's you. I think that's the feature we both see. Once I get this moving, we can turn to port a little bit.
Kind of peering into the darkness here, you can kind of see more of these coral colonies, those white skeletons really standing out. Wow, I think there's going to be quite a few on this pinnacle as we're making our way up to it here. Really large colonies too. See corals and sponges all on this pinnacle here up ahead of us. And yeah, looking down the wall here, that's really quite a sight.
It's about as vertical as you can get.
Yeah, I get a little glimpse of the vertical wall that we've just climbed up.
It's taller than 60 meters. We have no bottom lock.
Wow. Yes, the pilot's just saying this is taller than 60 meters. They're not getting a bottom lock on D2 right now or Sirius. So really see that steep face with all these little sediment chutes which have collected sediments and collected these coral skeletons as they've fallen down. It's really some massive colonies up here.
I'll note something that I had suggested we would see earlier in the expedition we hadn't, and that is that when you get these steep walls, very often the suspension feeders are at the top of the wall or near or approaching the lip of the wall, not on the wall itself. And that's exactly what you see right here.
That is a big sponge.
Yeah, it looks like a big, unfortunately now dead sponge.
Yeah.
This is one of the conolasma, and we're seeing the internal skeleton.
Yeah, it's really beautiful. Christopher Kelly suggests maybe it's an Africalystes. But it definitely looks like a skeleton that's been left behind. You know, it's missing that really distinctive brightness that you see in the live colonies. It'd be great to zoom in on that one. I see lots of other organisms using that habitat. You can see these little pink dots. These are snails. Yep, there's snails. Gastropods in here. This snail has its siphon sticking out. That's a little kind of straw-looking tube out of the top of that little snail. And you can really see that.
That tells you that.
Yeah, it's saying you can really see that beautiful texture. One of the pilots in an earlier dive had likened it to like a fiberglass sheet, and that's really what it looks like. A little hatching.
Do you see the coral in front of the snail?
Sorry, go ahead, Scott.
Just, for a second, the glass sponges, you know, the name Hexactinellida, the hexact refers to those spicules being six-rayed. So think of a cross then with two other points coming out in the other dimension. And they're all interlocked together, and that's what creates this nice web of fibers you're seeing. And yeah, there's a little soft coral there, it seems.
Yeah, a little octocoral.
Earlier, you know the snail is a predatory snail because of that siphon that's really well developed and sticking out, and it's kind of like a little nose that is waving back and forth and sucking water in and testing the chemicals in the water. But this may be the first truly soft coral that we've seen.
Yeah, this one you can't really see a skeleton through it at all. You can tell it's one of these octocorals from its 8 tentacles and the little pinnules.
The true soft corals don't have an internal axial skeleton, and so they are able to channel water into and out of the body to give them structure and support, but they can also force all that water out and so squeeze themselves down, just like an anemone would do in a shallow tide pool.
Yeah, absolutely. They have this hydrostatic skeleton. There's actually several of them as we back out of the sponge. I can count 3 of them, of those little colonies of those soft corals. And so those lasers are 10 centimeters apart. So this is a really big sponge, and really big structure for all these other organisms to live around.
Another interesting thing to consider about this sponge is, you know, one of the reasons it's brown is because, you know, some of the sediments that accumulate on it don't get moved off. Another is that it's, you know, tissue that's dyed and changed its color. But it's in such a contrast to the quote-unquote clean white sponges, which are constantly generating a current, and so there's constantly water moving around them, and those sediments and so on are being cleared off, which keeps that tissue white and healthy.
Good pilot.
Yeah, this is a really beautiful view here, too, of the kind of top of this little pinnacle edge.
I'm looking at you at 25 degrees, but you got some tether.
Copy that.
This is kind of a local little,
providing an
thing,
identification,
but
a possible
seeing over
identification
it.
to those soft corals that we're seeing.
It might be a little awkward as you pass over it, because it might go down afterwards.
Described family.
So we just have to see.
Copy. Looks like it continues a little bit over here to starboard, so I'll cheat to this side.
See quite a diversity of sponges and corals up on this ledge here.
But if you see, this is, so you're seeing all the way back, so yeah, we're approaching this step feature, so that might be real, we're not sure.
Copy that.
It looks like you're seeing...
Yeah, I'm seeing a little bit over there to starboard in the
blue view. It looks relatively flat right here.
Yeah.
If you want, I can move Sirius.
Yeah, I think
a little
so.
Closer that way. You want 15 meters at 270?
270 sounds good to me.
All right.
Yeah, it looks like there's some sandy features in the back.
Yeah, it looks...
I'll just kind of straddle this to starboard as we come around.
Sounds good.
So if you're just joining us here today, we have just under 25 minutes left of this dive. This is dive number 12 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. And this expedition has now landed us in the New England Seamounts. We've been in the Corner Rise Seamounts further to the east.
That's correct. Thank
and
you,
we're
Rich.
kind of stepping our way back into shore very slowly. The stepping stone that we're looking at today is a seamount that we've nicknamed Y Seamount because of a little feature on the very top of it. This was a previously unmapped and unexplored seamount before we came here. We landed at about 2,830 meters depth. We're currently at 2,589. We've been working our way up some extremely steep terrain, covered in lots of coral rubble, some fossilized, some new, as well as several species of coral and sponges, and some of the smaller fauna that we've seen today as well. So it's been a really exciting dive so far. Interesting seeing this channel in the middle of the top of this plateau, Kira. This is really, and it's filled with like sediments and coral rubble, and then live colonies on the sides of it.
Yeah, a really neat little feature up here. Hard to tell if it's from some kind of differential erosion here, or if it's a piece of it has fallen away from the other and created sort of a crack,
water column?
like you can imagine in an egg or something, and definitely sediment infilling in between. We've reached this little ledge along our journey up a steep ridge.
Looks a little bit different from the ones that we've seen.
Yeah, this one looks almost like it might be what we call a planktonic species, or a swimming that might spend most of its time up in the water column, though I'm definitely intrigued if anyone in the chat room, yep, so I have confirmation this is probably a planktonic species. And we can often tell that by the body being kind of clearer, more see-through. So these species spend a lot of their time up in the plankton. And we have from several people, Tomopterus is that type of polychaete that we just saw swimming.
How are we doing on samples, Nav? Sounds like we may have a suction sample. I know we got quite a few rocks earlier.
Yeah, we have 4 samples currently. We have 1 in our starboard inner bio box. Both of our rock boxes have samples, and then suction jar 2 has a sample.
Copy that.
And coming now back into this kind of sedimented region with more coral rubble, indicating that this platform maybe used to hold a lot of life, or maybe it's just accumulated. It's somewhere downslope where a lot of sediment and coral rubble from above has been able to accumulate on this little ledge that we've come across on our journey up this steep slope.
Yeah, it's really interesting, Kira. You know, this area, we see a lot of the live corals and sponges growing out of kind of the coral debris. And so I wonder, you know, how rocky is it underneath this sediment, or is the sediment quite thick, but these corals have accumulated and are stable enough that these other organisms have grown off of them?
Is complete.
It looks quite scraggly in this area.
Good word. I agree, especially compared to those beautiful, smooth, igneous flow features. Here's, a much more varied, scraggly landscape.
Absolutely. Absolutely.
And for the last 20 minutes or so, do you know what the plan is? Are we looking to just explore this area? Are we trying to get up to waypoint 2?
I think I was going to check in with Watchley, actually.
See more of these hemicorallium, the white and pink.
Break. Watchley, Nev.
Watchlead here?
Yeah, I wanted to check in and see kind of what your hope for the rest of the dive is. We have about 20 or 22 minutes left. We can head for waypoint 2, or if you have anything else you want us to do.
No, I think heading for waypoint 2 would be good. We'll just head as far as we can and then pull off when we need to.
Awesome, thank you.
Thank you so much.
All right, so just heading up to waypoint 2, which 270 is getting us there. So.
Copy that. You want to get another move in then? You think it looks pretty?
It looks pretty. You're seeing 50 meters out. I don't see anything.
Nope.
It's a little local high path here. I think we could do another 20 meters, 270.
Yeah, I agree.
Fine.
Let's go ahead and try and make some tracks while we only have a few minutes left.
Bridge, RV nav.
And here looking down where it seems like more sediment has accumulated, maybe even, and are we seeing little tiny patches of bioturbation? I feel like I'm seeing some lighter colored circles in the sediment. Not as striking as we had seen the other day
Go ahead,
on
sorry,
the
you're a little quiet. I'll turn you up.
Sorry, could you repeat?
But definitely looking like maybe where the sediment is thick enough to get some bioturbation.
That's it, thank you.
Is it right here in front of me, or I can turn a little bit to my port and get some of this here? Show our video if you want to snap zoom in on the sediment. We'll try and get the sand right where it hits the hard.
Kind of taking a zoom in here on the sediment, just to have a look and see what the composition is, and we've done this
come down a little bit
several times along the tracks that we've been trying to do during this expedition. And again, Kira, this looks really similar to some of the sediments that we were seeing early on in this dive, where there are some pteropod shells, you know, these white shells, but not as many as on other dives.
Slowly, video. I'll try to slow it down a little bit more.
Copy. Yeah, absolutely, and a bit more of the finer material that was absolutely absent out in the cornerized seamounts at the shallower depths, where it was just totally removed by the super strong currents. And here a bit deeper and a bit further in, we're getting some more fine sediments added into the mix. So likely a different infauna community living there, and giving us some clues about the current speeds and what's coming down as marine snow from above in the water column.
Actually back in tight. I think there may be a little thing in there.
Yeah, it was an interesting thing to note, Kira, that the currents are very different here than what we were experiencing on the corner-raised seamounts. On the corner-raised seamounts, we had currents, coming up to close to 2 knots.
Too many of them this cruise.
And here we had basically nothing this morning. There's this beautiful upside-down squat lobster hanging upside down from this branch. You can see its long, the long pointy thing on the end there is a part of its rostrum, which is kind of like a nose on the end of these squat lobsters. You can see its legs kind of dangling on. And so these squat lobsters, are picking particles out of the water to eat. We see them on the rock faces. We also see them on live corals, and here's one hanging out on this dead coral skeleton. Yeah, and these thicker sediment areas.
Yeah, are you seeing the little bioturbation patches that I'm seeing in this little sediment pond here, where it looks a little bit lighter color? Just guessing why we're getting sort of spotting in this little sediment pond here.
Yeah, absolutely. You can see those lighter patches of sediment where the sediment's been turned over, and that can be from. Lots of different organisms can do this, and we'll dig burrows. There we are. We're actually going to zoom in on one there. And so this is from organisms, digging into the sediment and bringing up sediment that's a bit deeper and bringing it up to the surface, and then also bringing that surface sediment down to the bottom as well, and potentially feeding on some of the food that's been landing on these sediments. And these can be worms, polychaetes. They can also be things like amphipods. Lots of organisms burrow into sediments, urchins as well. Brittle stars. There are lots of animals that live inside sediments or live around the sediments, and will be able to turn over sediments like that.
Give us a little tilt up, looking in the distance.
Brian, just coming up ahead on the left, maybe it's not so much ahead anymore, but there's a whip. If we can have a quick look, if possible.
A whip coral on the rock.
Copy that, thank you, Bridge. Ship move is complete.
Understood now, thank you. Whip coral on the rock.
Yeah, where, Scott, could you give us a bit of a description?
Still left out of frame on the other side of the channel.
Oh, left out of frame, okay.
big upper left.
There. Ah, it has branches. There you go. Yeah, could we have a zoom in on that coral there?
Oh, look at that.
Some strange branches.
Well, I'm even more intrigued.
Go ahead and start coming in. Yeah.
Great colony in the background there, though.
I know. These colonies are absolutely huge up on this pinnacle. Yeah, this is really interesting. It's got 3 little scrawny branches. This bamboo here.
It's a bamboo coral.
Yep.
For sure.
Coming down.
And it's a nodal branching bamboo coral. How about that? Okay.
Yep.
Thank you.
Yeah, you can very clearly see the branches coming out of the nodes of this one. So these bamboo corals are given their name from these, what we call nodes, these black areas or sometimes a dark brown. And these are made out of proteins, whereas the rest of the white skeleton is a calcium carbonate.
Oh, yeah, I think so.
And so they
note how transparent the tissue was. You can see that skeleton very clearly. So this is very different from that much larger Chase Nisus type nodal branching colony that we saw deeper today.
Yeah, and there's actually another one of it to the left there as well.
Pretty sure I've seen over it on
you saw that,
but I just want to verify real quick.
I did. Thank you. So is it? I think that's a primella, possibly.
Back up again.
That really massive fan.
Yeah, the massive one on the left?
Because of the curling, yeah, the massive one on the left because of the curling branches.
So you're seeing 20 meters out, which is good for you. So it's probably 25 meters from me.
5 or 6 balls.
Pilot, this is watch lead.
Go ahead, watch lead.
Could we zoom in on the really big coral on the left?
Yeah, the one coming into center.
Yeah.
Sure.
Thank you.
Camera down
over there.
Oh, those are the fishing brittle stars.
They are. Yeah, I think.
Almost assuredly a primnoid, and I'm guessing that because of the curving nature of the branch, and now because of those fishing ophiuroids, which we never see.
We'll try to start off with a full screen.
Another example of where the
sounds like this is of interest. Let's get a good tone.
Beautiful colony.
Come down a little bit.
As close in as you can get.
Copy that.
Yeah, and so Scott France was saying that these are fishing ophiuroids, and so these ophiuroids are picking particles that go by out of the water column. And so you can see their arms sticking out of this coral. So the coral has managed to get them up off of the seafloor and into better flow, and then they stick their arms out with the current, trying to really find food and bring it into their mouths.
Yep. Copy that.
There are a lot of people down here in the Louisiana Bayou who could really appreciate those ophiuroids, dipping a line into the water.
Right, yep.
Fishing for their meal, yep.
Yep.
Okay.
Some more of these
at
colored
the base.
hemicorallium as well. Yep, at the base and work our way up is great. Yeah, looks like there was another big branch there at one time that's broken off. Quite thick. And interesting how it's settled right next to this this more fossilized coral branch that's here.
Right up.
So you want?
Yeah, settled next to it and kind of growing up on it or clutching onto it, using it as stabilizing its own colony.
And let me know when you want to continue.
Go ahead.
Coming up.
Like some new growth polyps that you can see coming out of the tissue there, and this
nice,
is another thing that we've talked about in the past.
branches.
Even though
start coming in
we would interpret the base of the coral, of course, being older than the tips of the coral, it doesn't necessarily mean that the polyps in the tissue itself are that old, because it could be replaced through time.
Right, right. These little upward-facing polyps. Some are open, some are closed.
And the orientation that you just noted facing upward, the orientation that they can contract, whether they're contracting upwards and towards the stem or downwards and towards the stem, or if they're more or less parallel, is one of the characteristics that we use to identify it. So that's a good observation right there.
Yeah, and then Tina and Chris Kelly have also mentioned that there are actually some of the polyps that are facing downwards. In this screen in particular, if you look either dead center or slightly to right, you'll see some of those polyps are actually facing downwards and some are facing upwards, which is really curious.
That is not helpful at all.
I thought you'd say that.
Oh, geez. There's a nice little mysid, a possum shrimp, on the far right side there with the bright orange eyes.
Yeah.
On amphipod, which is a close relative, different order, but a close relative in the crustacea that was lower on the colony facing downwards.
Yeah, look at that. Polyps up, polyps down.
Hold there.
Hold there.
Steve Cairns is going to shudder about this.
I know.
No.
Continue up.
Coming up.
The possum shrimp are so called because they carry their embryos in a little pouch just underneath the thorax.
Yeah, there's a few of them on this colony. Those bright eyes staring at us.
Copy. Coming up.
Yeah, it's very interesting. We see a lot of mysids often associated with coral colonies. Seems like a good place to land. You know, we call them shrimp, but they're not closely related to the true Penaeid and Caridian shrimp that we see.
Continuing.
Shrimp is kind of a term like coral. You know, it's descriptive of a body shape that has many legs and is elongated. And
looks like there's one up a little bit further.
But it's not a taxonomic
kind of just the brightness there.
Group of organisms that are closely related.
How do you feel,
Right.
co-pilot?
Straight up.
Some of these brittle stars have had something crop their legs. Is the one on the left here is missing parts of two of its legs, which will grow back.
Okay, I'm going to hold
and this
there
one
for
on
a
the
second.
top here too. Oh, beautiful, really beautiful. And I believe these are they an astroschema? I'm kind of asking the chat room here or Scott.
Can you center that guy?
Yeah. Coming over.
Ready when you are.
Coming.
I should remember the name, and I do not at this moment remember the name.
That's okay. There's a lot of names to remember.
Bail us out.
Yep. Beautiful. And you can actually see on that uppermost arm
Coming
where
down.
it's starting to regrow its tissue.
Max soon.
Copy. I'm going to hold
So these
there.
brittle stars are echinoderms, so they have what we call pentaradial symmetry. So they have symmetry in fives. You can see those 5 arms and the 5 little triangles and its disc there in the middle. Really beautiful.
Just tilt up to the top of
The other thing you can say
arm.
about brittle stars is that their arms are packed with
Hold there.
ossicles. Ossicles are the small microstructures that are
Pulling out.
calcareous that are embedded in the tissue of echinoderms.
I'll
that
start
give them the
tilting
name
down
finer
once I see the
skin.
top of the
But in brittle stars, they're oriented
coral.
into these vertebral-like
Okay, let's
see if we can
structures,
show
same sort of
3 to
thing that
4
make up
of the
your backbone. And
ophioids.
that interlock and orient with one another all the way
Go
along
down a
the
little
arm.
bit
And
more.
what makes
Coming
the
down.
arm so
Maybe push left a little
flexible
bit.
is the
Push
connections
left.
between them. There's some
As far as
musculature.
rotate.
And one of the
Rotate
really
left so we
fascinating
can see
things
the
about a lot of these
ovoid center.
echinoderms is they have what's called mutable
Coming down.
collagenous tissue. And so the collagen, the connective tissue
pretty good there.
between those structures is under control of the nervous system, such that it
Holding
can
there.
become more solid or more liquid under control of the nerves. Just fascinating, and it's a way of conserving some energy so that once you get the arm in the position you want, or a stalk in the case of a crinoid, you can kind of fix it in that position.
And we'll go
Yeah, the
full
other thing
screen.
that these brittle
Copy
stars
that.
keep in their arms, kind of in their upper arms near
Tilt down a
the
little
disc,
bit
is
as you
actually
pull
the
out.
gonads. So that's where you'll find the eggs and sperm and where reproduction happens in these brittle stars. They don't really have a
Hold
lot
there.
of room in their
That is
bodies, and so
full
they keep
wide.
them
Copy
kind of
that.
just in the very tops of their arms near the central kind of pedal disc.
It'll lift off and sort of back up and then
Yeah, I'm going to
rotate
lift
it
off and then try and like tilt down
And
as
so
I
Christopher
come up.
Ma has come in and said, yep, astroschema, perhaps. Beautiful, really beautiful colony.
Nice
Thank you
and
so much for
slow.
that, pilots.
Pilot C was cam 6-5, leave a nice delta.
Rotate back to wherever. Continue the
that.
expedition.
Copy. Continuing expedition. I'll push out for you, co-pilot.
Copy that. I'll follow you up the camera.
Stood. Come back to center. And push it forward.
Camera's at 4-5 right there.
Understood. Thank you.
We have about 7 minutes before you want to set up.
Copy that.
That's giving you 5 minutes for setup, so.
Understood. Should be a relatively straightforward one where you're pretty much almost lined up with the ship. As it
Yeah,
is.
could probably just drive
Seems like we're getting back above the little platform and starting to get a little steeper here. Still seeing these igneous flow features, lots of sediments cascading down.
Camera's 3.5 right there.
Understood. Thank you.
Yeah, this kind of looks a bit more pillowy for the underlying with some of those areas where it's caved in. You can see some of these kind of little amphitheater type morphology in the seafloor pillows there.
All these little bumpy pillows of lava, mini pillows. Very cool morphology with those vertical abrasions on each pillow, creating this funny striped texture and when the sediment fills in between.
Do you mind doing a quick snap zoom here, video? Center, white, just look kind of odd.
Yeah, beautiful. It's a sponge with a crinoid on top, and it's kind of arched over. Really beautiful.
Pilot's clear when video is. Just a little different.
And that huge, nearly intact dead organism covered in that ferromanganese patina, it's not really broken up very much.
It's amazing, isn't it, that I've seen a lot of those as we've gone on this dive, that they're not like broken little pieces like we were seeing in previous dives. These are like whole colonies that have come down in one piece and have actually stayed in one piece.
Yeah, I guess suggesting that at those shallower depths there was a lot more activity, a lot more high energy environment, maybe even some wave action that had broken up the coral into pieces. And here, in the deeper depth, these are the ones that were able to fall and get coated in that ferromanganese over tens of thousands of years. More intact. Very interesting. Nice igneous features here too. Always thinking about the many rocks we can pick up and look at and see what story they're telling us here as well about the formation of this interesting, previously unmapped and unexplored seamount.
Yeah, he's one of those beautiful sponges that were actually, this looks very similar to the sponge that we collected as kind of our first thing this dive when we were down on those sediment waves. If you remember, Kira, there was just a couple of these large sponges that looked just like corals, and here we are, several hundred meters higher, and I think they're here again.
Two of them, maybe two separate ones here.
Yeah, the Stellodorix sponges.
Kind of neat how they're both delicately balanced on that skeleton.
Just perched, yeah. There's another one on the rock as well.
Yeah.
Look at the one.
Yeah, it's amazing.
Right.
You know, we saw those first at 2,830 meters. We've made those first collections, and yet here they are at 2,581 meters. The depth range of some of these organisms that live in the deep can be really, really large.
Pan up a little.
A little party going on here. Lots of different stuff all together.
There is, yeah.
Pilot's clear when video is.
Clear.
Yeah, and these beautiful igneous flows covered in ferromanganese crust, looking a little bit less fresh of growth. You can see that instead of being that really true black color that indicates freshly precipitating crust, these are covered with a thin layer of sediment that's probably slowed or even caused a hiatus in the growth of the ferromanganese crusts. where we're seeing this little bit more higher sedimentation rate.
Yep.
Some of these stalk sponges that were so prolific in some of the corner-rise seamounts, and I believe this is the first one that we've seen on this dive so far. So really curious.
It's also hard not to see some of these eroded rock faces and think, oh, I wish you could just grab a rock. But so often, even though they look like pieces, these are actually still stuck in place. And we've had 2 nice rock collections earlier in the dive today.
Yeah, it's definitely hard.
Probably want to start
Kind of
setting
looking
up
down
for
the
off
face
bottom
here
soon.
across to the side of us.
Copy that.
See more of those.
Copilot, feel free to
More
start
of these
stowing lights
corals.
if you'd like.
Copy that.
Watch lead, this is pilot.
This is watch lead?
We have about like a
Yeah,
minute or
much-
2 left on bottom. Anything in particular you want to see in this area?
No, I think we're good. Thank you.
Understood. All right, we're gonna have Copilot start stow, and then we're gonna go ahead and get ready to recover, or at least start ascending.
Go ahead, Kira.
Oh, sorry. I was just saying I panning back and forth between these two. It's just interesting seeing this much more rugged looking and broken
maybe come
pieces
in tight
on
on
this
this,
side, and then we were
crack
just panned over
roller.
to the right, the more intact flow features of the igneous rock. Super interesting.
How are we looking, Copilot?
All swing arms.
Yeah, this is an
are
underhang
still.
here.
Copy that.
Overhang, underhang.
Front packs off. HB off.
Thank you, video. When you're clear, we'll go ahead and start recovering. All right. You want to come up with me a little bit?
Thank you so much,
Copilot?
pilots.
Absolutely.
All right. Coming up. Once we get up a little bit, I'll have you put an auto head.
So with that,
And
we're
then
going
I'll,
to leave the
already in.
seafloor. So that is the
Copy
end
that.
of Dive 12 of the NOAA Ocean Exploration
been
Expedition,
interesting.
North Atlantic
Wow.
Stepping Stones. I want to thank
I'll go in auto
everyone
head
for
as
watching
well.
with us today.
We'll start driving backwards.
And
Copy that. Still
on behalf of myself and
Good.
everyone on board here on
I'll
board
stop.
the Okeanos Explorer, and also all of our scientists
Going
from
out of
shore,
depth here.
I hope you will
I'll
join
start
us
coming back.
tomorrow.
If you want to come up to a Delta 18 or so.
Sounds good. I'll follow
Another
you
great
back
day
with the camera.
exploring the bottom of the ocean. Thanks
that.
everybody on board. Thanks everybody at home.
Delta 14, 15 there. I'll grab some more.
Come back a little bit quicker now that you're getting up there.
Thanks so much, everyone.
Looking good. Alright, it's Delta about 20 right there.
Understood. You got me at what, 7.5? Come down to till I'm underneath you when you're looking 90, and then I'll go ahead and turn to starboard.
It's
Almost
80
180
there.
degrees. Then I'll push out 85. I'm gonna start rotating.
It's 90 right there.
Starboard.
Tether looks good, flying right over.